怎么突然又不行了？哦，啊，哦，啊，我
Hello.
Hi, Marco.
Hi, how are you?
Yeah, I'm fine. How are you?
Good, good. Excited. Where are you connected from?
Oh, actually, I'm holidaying in Seville, Spain.
In Sevilla. Oh, nice. Is it good weather at least?
Yeah, very good weather.
I'm in Cardiff and it's raining.
And where are you?
Yeah, I'm in Cardiff. I'm in Wales. Everything is locked down in here, so can't go out much. How's the situation? Do you usually, are you from Spain? Do you study in Spain?
Yes, in Basque Country.
Basque Country.
Yeah. So
Okay.
they have now closed the border, so we are going back in 1 or 2 days.
Right. So there is the border between the Basque Country and the rest of Spain.
So there are like a few autonomous regions, they have closed their borders due to this COVID situation.
Sure.
Yeah.
Yeah. No more holiday.
Yeah.
Hello.
Hello.
Hey, this is so cool. Okay. I'll turn this up a little bit. All right. How are you guys doing?
Good.
What time is it in your location?
It's 12:52 p.m.
Yeah, same here.
And in Cardiff, it's 11:52.
All right. Yeah, it's early morning here, 7:50.
Oh, okay.
I hope my kids won't come up and bother us.
Where are you connected from?
Toronto.
Oh, okay.
Yes. And did you meet Lucy yet?
No.
She's gonna be your support, technically. So she's in Beijing, so it's 8 o'clock in the evening there.
Lucy is here.
Okay.
Yeah, she's Zoom room 9. Okay. Now let me see.
Daniel, where are you connected from?
Here in Oslo, Norway. Marco.
Oh, okay.
And I might have missed it, but where are you at?
I'm in Cardiff, in Wales.
Cardiff. Yeah. And Dipanjan?
I'm at present at Spain, southern Spain.
I think the second I came in, I heard Basque Country. Are you in the Basque
Yeah.
Country?
No,
Yeah.
I'm in Seville, but yeah, my institute is in Basque Country. Yes.
That's okay, Lucy. Thank you. Did you manage to set the YouTube link? Oh my God. I hope you can hear us. Okay. So Daniel or Danny, can I say Danny?
Danny's great.
That's good. Okay. Just call me Marius, right?
Okay.
All right. Okay, let me see. I just got your email. I just got up, and so I just got your email. Let me see. That stuff in there. Right. So you're going to go first.
Great.
Am I right that you attended Neuromatch Academy?
Yeah, that's right.
Cool. So you know who I dance with.
What's that?
So you know who I dance with.
Yeah, that's right. It was a great, great intro to this computational world.
Yeah, I'm not that computational myself, but
Okay.
I might like be a student next year. So for everybody else here, by the way, including Lucy, the Neuromatch Academy thing, it's a grassroots summer school, but like 8,000 people participated with like over 200 organizers, and it was great. Right, Denny?
It really was great. It was awesome. I was very impressed with.
Sorry, Marius, I couldn't open the YouTube live now. Maybe you can help me.
I no longer have the ... menu here.
Is it okay? Oh, you don't have the menu too. Okay, wait a minute.
You have to make me co-host. Can you make me co-host?
Do you find it? It's in the more, under more. Sorry?
I don't have more because I'm not a co-host. Can you make me co-host?
Oh, you are a co-host.
Oh dear.
You are a you are not a co-host. I set you as a co-host. Maybe you can be a host, but I think it doesn't matter.
I'm the host now. Okay. Well, first of all, let me do the official background. Yeah. Virtual background. Okay, fine.
It's okay.
I was just setting my virtual background. There we go. I have to do, now I have the more menu. Okay. Stop live stream. It already, I can only stop the live stream there.
Sorry?
I can only stop the live stream there.
You stop.
Do I stop it?
You mean stop?
My menu only has stop
Okay.
live stream. I cannot do the other things.
Okay, maybe I try it. Maybe I try it.
Marius,
Yes.
I've slightly changed the title of my presentation. I don't know if you.
Okay.
So
In the chat.
I'll just put it in the chat.
Oh, Lucy, it is working. Dimitra can see us.
Oh, working.
Yes.
Okay, so you find a customized live stream, the
No.
menu.
No.
What menu do you have?
No, it was already working. So Dimitra
Already working.
Dimitra checked it.
Okay.
YouTube
Okay.
stream and she's seeing it. So we're live.
Okay, maybe.
Okay. So Danny's gonna go first, and then Marco has a slightly changed title. Let me put that in my.
Thanks.
And Thipanjan is gonna go second, and then very cool. So you're all doing fMRI, right?
Yeah, I'm doing it. Right?
Yeah, but I'm not presenting any fMRI results.
No. But it kind of makes sense that you're in the same session. So did you guys read each other's abstracts?
Yeah, superficially.
I thought so. So I hope we're going to have good discussions, even if it's just the five of us. Okay. Let me see, do I have my notes here too? Yeah. That's maybe better. I have two laptops. So I can have the notes on a separate screen. Hey, so Lucy, when we open up, can you put in the chat and ask everybody in the audience to put their name and say where they're from? I think, like I've seen one MC do that, and it really got people to talk.
Okay. No problem.
Because other times, and maybe it was because it was too late, but I found that as audience, you don't see anybody else except the speaker, and it can be very lonely. So having a bit of a chat, just get that going with something stupid is probably a good idea. Okay. Okay, Lucy, can we open up the room for everybody? I think it's time. Cool, thank you.
Marius, you are the host now. You should open it. You should open the room for the guests.
How do I do that?
Can you?
How do I do that?
Yeah. You can put me as host and I do that.
Oh yeah.
Change me to host. I think there is, yeah.
Can you make me co-host? There you are.
Okay. Okay. Now they can come.
Let me just connect it.
Yeah, cool. We got 5 audience now.
Oh, lots. Interesting. Okay. So we're going to have our session this morning with 3 talks. First, we were going to have Daniel Brown, or Danny, talking about resting state fMRI, connectivity in schizophrenia, bipolar disorder, and healthy controls. We're going to have Dipanjan Ray, who's going to be investigating intro and exteroceptive predictive processes in major depressive disorder. And finally, Marco Bigica from Cardiff University on selective impacts of sleep deprivation on brain and behavior. I'm a behaviorist, so we want to see that. So I'm Marius de Hart. I'll be hosting the session today, and on the back end is Lucy Liu, organizing all the technical stuff that I have no idea how it works. Okay, so to begin with, we're going to have Daniel Brown. And I'm going to give you his Twitter handle so you can spam him afterwards with all the questions that you didn't ask here. So he's from the University of Oslo. And he's going to be talking about resting state fMRI connectivity in, thank you, connectivity in schizophrenia, bipolar disorder, and healthy controls. He is a master student in Lars Westlight's web, and he was a student in a Neuromatch Academy. And for everybody here who didn't do that yet, next summer, be a student at NMA. It's going to change your life. Okay, Danny, take it away.
Thank you, Marius. Thank you, everybody, for being here today. As Marius mentioned, I'm a master's student. I just started learning computational neuro, so would love to get everybody's feedback and just here to learn as much as I can. So the title of my talk is Resting State fMRI Connectivity in Schizophrenia, Bipolar Disorder, and Healthy Controls. And I'm a master's student of cognitive neuroscience here at University of Oslo. Working with Håkon Engen at Norment, which is the Norwegian Center for Mental Disorders Research. And I'll just dive right in. So we're looking at connectivity analyses, which I'm currently learning. And so the gold standard, or what we're really looking for in this, is biomarkers for mental disorder. And why that might be something we really want to find is because this could improve the reliability of diagnoses of mental disorder. For example, if we could just use a method like EEG or fMRI and kind of peer into the brain and see how everything is working, then we could understand maybe this person, we could make differential diagnoses. And increasingly, we're starting to realize that within mental disorders, they're not all the same, meaning that there are distinct subtypes of many mental disorders, like, for example, depression and maybe schizophrenia as well. So this opens up a translational impact where if we knew what type of subtype of, say, depression or schizophrenia one person has, you could map that to the ideal treatment for, that we've determined through basic research. And increasingly, I'm beginning to, and I know that a lot of other people are starting to think of these mental disorders as circuitopathies, meaning that distinct circuits can be implicated and lead to these different subtypes of mental disorder. So how do we do these connectivity analyses? Well, in the research, we use these graph theoretical constructs where we have nodes that are usually parcellations of the brain, and then we have the connections between those nodes, which are the edges. And there's different types of measures of this connectivity. We can have effective connectivity where it's directed, meaning we have a parent node and a child node, and that edge is a connected, directed, a directed connection between those nodes. Or we can also just have functional connectivity with undirected nodes, basically just correlation between nodes. And there's been all types of computational methods to look at this, like Granger causality, dynamic causal modeling, and each one of them. Yes?
We can only see your first slide. Sorry to interrupt.
Oh, I'm so sorry.
Maybe you're sharing the wrong screen. I don't know what's going on.
I'm not sure either. Oh, it says sharing is paused. Okay. I had some type of issue here. Just 1 second.
Don't worry.
Are you able to see my screen?
That is better.
All right. So, so I've been talking with just the first slide, huh? As I said, the gold standard here is we're looking for biomarkers for mental disorder, thinking along the lines of circuitopathies and graph theory. So we've got all these different connectivity analyses like Granger causality, DCM, and they have common pitfalls and they also have common or different benefits, each of them do. So we wanted to figure out which one should we try to use. And the one that we wanted to use is a relatively new one called the DGM method, which is a dynamic graphical model. So the big paper here is by Schwab and colleagues from 2018. And they describe this model and also try it on some ground truth data. They made some synthetic neural data and then tested this model out compared to other connectivity analyses. And this is a model for directed relationships, so directed connectivity. Without getting too into the weeds here in the interest of time, one of the things that DGM does, which is shown at the bottom, is it compares different models of connectivity. So it compares bidirectional connection for each pairwise node combination. So from A to B, from A to C, from all the different nodes. And it tests it for being a unidirectional from A to B, or a unidirectional from B to A, and it also tests a bidirectional. And then it tries those out and using a log Bayes factor, picks which one kind of wins in this battle and then assigns that to the model in an extremely simplified method of explaining. So what we wanted to do is use our data that we have at Norment. This is the top data set, thematically organized psychosis research. This was collected beginning in 2002 in 4 major hospitals in Oslo. To the bottom left, you can sort of see the breakdown of these with how many individuals with bipolar disorder diagnosis, schizophrenia, healthy controls, and other. We also used some data from a stroke sample that was collected using the same paradigm to get some additional healthy controls in there. You can see to the right-hand side on the bottom, we have our image acquisition specifics, where it's a 3 Tesla scanner, 3 millimeter voxel size, and we're looking at resting state fMRI data. The TR is 2.25 seconds, and there's 200 total scans for each individual. So individuals are sitting in the scanner, just kind of mind wandering, and that's the data set with 200 time points, 2.25 seconds apart. So our very preliminary findings, just to show some data, to the top right, you see our top sample in the DGM method. So the way to read this is on the left-hand side, we have our parent nodes, and on the bottom, we have our child nodes. So that's the directional, the direction comes from the parent to the child. And the first thing you'll see is that we have really strong connections bilaterally. So from the right BMPFC to the left, and from the left to the right. So that's what these diagonals are. But before I go in and look at anything else from this, this model, I want to point out what I have on the bottom right here, which is our average discount factor. So this is a parameter within the model that measures the variability of the nodes and their variability across time. And when this number is really close to 1, it means that there isn't any variability if it was a purely 1. If it was 0, it'd be completely uncorrelated and just noise. And through some conversations with our colleagues, we've heard, and they've run this on other data sets, this parameter is usually in their data set was about 0.8, 0.7. And so we're seeing from this that there isn't a lot of variability in our data set, and we're kind of trying to understand why is that. So the limitations that we're bumping into now is that we have a small number of time points in the sample. So, and it might not be enough to do this kind of method. There's only 200 time points in the sample, and then the TR is also very long. So that's a time in between the scans. So if we're talking about these, the dynamics in this system, in a neural system, and we're taking measurements 2.25 seconds apart, are we really going to be able to tell anything interesting, or does the brain act faster than that? And other things that are important, like I said, the variance in the nodes, maybe we just don't have enough variance in the data, maybe we need more data. And there's also, motion can also be interesting. So, and I should say, we reran this. We originally had our data thresholded for noise at a certain threshold, and we turned that threshold down, thinking that maybe we'd introduce some more variation into these nodes, and that did not, change our discount factor, move it further away from 1. So with that, I want to leave as much time as possible for us to talk about this. Does anybody have any ideas where we should go with this next? Our first ideas are to try either a new method with the same data set, or probably more likely a different data set that has a shorter TR and maybe more actual scans, more time points. Great, thank you guys.
Oh, that was awesome. Okay, I don't see any questions yet in the Q&A or in the chat. Wait, let me scroll in the chat, see if I missed something here. So people, start typing your questions. Oh, Marco, do you want to ask a question? Go ahead.
Thanks, Daniel. Very nice presentation. I'm sure you said this. I probably missed a part with, but so you said that you're looking for biomarkers. So of these conditions, and I'm not sure I understood how this directionality can act as a biomarker.
Mm-hmm. Yeah, sure. So, for example, the only thing that we're showing or that I'm showing in this graph, and that that's because of the shortened time period, is this is everybody put together. This is the entire top sample. But if you were to look at this same model of across group differences, right, where you have healthy controls graphed in one, schizophrenia on another one, and bipolar disorder on the other, you might see this different pattern where maybe this connection is really strong in schizophrenia, or is like flipped in bipolar disorder or something like that. So what we do is we take these models and then we do group differences, and we look at the differences between them. We compare them.
So this is currently based, this N1000 this is what sample? Sorry.
Yeah, this is on the bottom.
The total sample. Okay.
Yeah, and it's just from top in this scenario. But that includes.
So it combines both healthy controls and patients. I see.
Mm-hmm. Yeah, and because of this discount factor, we kind of like haven't even been able to progress to what we really want to do, which is the group analysis, where we start to answer that question that you were asking.
Okay, Dipanjan, do you have a quick question?
Yeah, so I have a couple of questions. So I think I can read the paper about this particular method you are describing, but from this from this present slide, so you mentioned the present slide that you presented, the result. Can you go to the result, please? Yeah. So as it is, I understand it's a effective connectivity. Is it effective connectivity? Like, so then why this connectivity appears to be symmetrical, like this matrix? Like I mean, the connectivity, which is, so can you please explain on those things? I mean, of course, I don't know much about details about this particular method.
It's a very good question and something I hadn't considered, but the first thing that I can think of is that it is directed, but it doesn't seem that it's limiting to only one direction, if that makes sense. Because like I said earlier, it's comparing these three different models. So it could be a bidirectional connection. It's also compared to unidirectional from B to A and one from A to B. So yes, it could be the case that A to B, a unidirectional wins, but also a bidirectional could exist. So I think that's the answer, and that is my work right now. Yeah.
So here in your data set, you mostly find bidirectional connections, you mean, like which is similar.
Yes, if we were to like interpret this, I mean, well, you know, the strongest ones are these bidirectional ones, and that's exactly what you will see in this model all the time because they're the two hemispheres of the brain. You know, they're corresponding regions. But then there are also all these other connections that show up, but really we're not even interpreting this because of this.
Mm-hmm. Yeah.
Very fascinating. But we have to move on, unfortunately. Daniel, can you unshare your screen so that Dipanjan can start? And I will announce him. So Dipanjan Ray is a postdoc with Rem Frost in the Basque Center on Cognition, Brain and Language in San Sebastian, Spain. And he's investigating intro and exteroceptive predictive processes in major depressive disorder. Sorry for mangling that. Take it away. You're still muted.
Can you hear me now?
Yep.
I lost it. Just a minute. Oh, I cannot see the screen.
You cannot see your screen either.
Okay.
Here again.
You cannot see my screen, no?
No.
Now?
Yes.
Okay. Oh, sorry for the confusion. So thank you. Betraying the original title, I am also like mostly going to present the exteroceptive part of our analysis, and it's because we found some bug in the analysis in the interoceptive part. We're just now modifying it, and it's very early stage, this present work, so I'd like to have your feedback to develop further. So the several alteration of the sensory motor function is known in depression. Overall there is reduced sensitivity of sensory perception, and there is also motor retardation leading to reduced speed and delayed motor initiation, slow speaking rate, and loss of facial expression. But please note there is a lot of heterogeneity in these things. So like in different subset of depression patients. So there are a few studies investigating the neural underpinning of these changes in the sensory motor function in depression. But our understanding of the cognitive process in the brain is undergoing a rapid change. So in contrast to the conventional bottom-up conditional approach to brain, which envisages brain as a passive organ. There is this related theoretical framework, say predictive coding or active inference, which envisages brain as actively generating prediction about the external world. And according to these frameworks, the core mechanism, organizing mechanism of brain is to minimize the prediction error. And it is subsumed in two different ways in sensory and motor domain. In sensory domain, the minimization of prediction error is subsumed by updating the prediction about the external world. So here, according to this theory, so the top-down connections carry information about the prediction. It inhibits or explains away the incoming stimuli in the lower cortical areas. And then these bottom-up connections carry this prediction error to update the prediction of the higher cortical regions in the sensory domain. On the other hand, in the motor domain, the minimization of prediction is carried not by updating the predictions, but by, in a way, fulfilling the predictions. So it is by actually acting on the external world, so that and changing the external world so that the incoming stimuli cohere with the prediction. So the predictive coding theory in motor domain is not very precisely defined. So I take this liberty to describe what I think should be the nature of the connections in the motor domain. So here, the top-down connections mostly carry the predictions about the external world, but this should be facilitatory, because ultimately it leads to the unlike in the sensory domain, because it ultimately leads to the activation of the lower motor neuron. On the other hand, the bottom-up connections mostly carry the incoming information of the sensory world, just like the sensory domain. So based on this, so our present work, we implemented the effective connectivity analysis employing spectral dynamic causal modeling on resting state fMRI data from depression patients and age and sex match healthy control from an open data set. We extracted time series from the important relevant regions involved in the sensory motor functions in the brain. And our output is the mean effective connectivity in the different brain regions, hierarchically organized brain regions. The mean effective connectivity across depression patients and healthy control, and also the group difference in effective connectivity in healthy control and MDD patients. And here is the, so here is the nodes based on the hierarchy. So the nodes we selected for that, at the topmost level, there are frontopolar regions, which is situated the highest order in the hierarchy. Then there is DLPFC and VLPFC. And then there are lower cortical region involved in sensory and motor processing, like primary visual areas, primary auditory areas, somatosensory cortex in the sensory domain, and supplementary motor area and motor cortex in the motor domain. So this is the results of the mean effective connectivity in MDD patients and healthy control. And here the red connections denotes the effective connectivity, which is facilitatory, and blue denotes the inhibitory effective modulation in the left and right side. It's congested. But I think it is apparent that the net top-down in the sensory domain, the net top-down connections are inhibitory, like from FP1 to primary visual cortex, or FP1 to somatosensory cortex or auditory cortex. On the other hand, the net bottom-up connections are mostly facilitatory, which is compatible with our conceptualization. And also in the motor domain, the opposite, like the top-down connections are mostly excitatory. So this is about the mean, and bottom-up connections are inhibitory. So this is about the mean effective connectivity across the MDD and healthy control. Now coming to the effect of depression on effective connectivity of resting state data. So this is the connections which survives at the group level. so this is significantly different in MDD patients and healthy control. The red connections denotes the connections which are increased, and the blue connections which are reduced in MDD patients compared to the healthy control. So we are still to come with a coherent explanation about all this observation, but there are some observed connections which goes well with the symptomatology in depression patients. So for example, there are increase in connectivity strength in the sensory domain from FP1 from the higher to lower regions, like FP1 to primary visual cortex and FP1 to primary auditory cortex. So there, in other words, there's this disinhibition of these top-down connections. So which may explain the reduced sensitivity of different sensory perception in depression. Because so if you see in the mean effective connectivity, there is these connections were inhibitory, and here it is positive, means it disinhibits this prediction. In the motor domain, on the other hand, important findings are like bilateral self-inhibitory loop in the supplementary motor area, so which may in a way explain in depression patients, explain the motor retardation in depression. But to be frank, we are still to explain all these connections and their neurophysiological significance. And of course, this study has many limitations at this present stage, because we have not considered for example, the effect of medication. So we are going to incorporate the effect of medication in analysis in the PEB, in the regression analysis, and also we are planning to implement this analysis on another pool of subjects who are like medicine naive. And also, as we all know, all this depression is a heterogeneous condition. So so we are also going to consider different dimension of depression and how this affect this affective connectivity in the different hierarchical region in the sensory motor, like both interoceptive and exteroceptive cortex. So yeah, this is all about our presentation. So I'd like to have your opinion and criticism so that we can develop our work further.
That's amazing. Thank you. Again, I've been looking at the Q&A. People, put your questions in there. Oh, here's a question in the chat from Ingun. I'm taking an intro class to neuropsychology, so I don't understand a lot of what you're saying. Could you sum up your findings shortly in simple terms?
Okay, so yes, of course. I mean, this is my first time to present something in 5 minutes, so I'm sure that I'm confused. So if you ask me to sum up, so we investigated how the top-down and bottom-up connections in the different brain regions involved in the sensory motor functions in the brain changes in depression patient. And we found that overall there are decrease in the top-down inhibitory influence of the higher order cortical regions to lower order sensory regions, and also there are more self-inhibitions of the supplementary motor area. So this may be one. Be underlying. This connectivity, we are trying to connect it with the depression symptomatology of reduced sensory sensitivity and motor retardations in depression patients. So this may be a potential neural underpinning of these symptoms.
All right. Ingun says, thank you. Hope this helps. So Thipanjan, there's another question in the Q&A. I hope you can answer that by text, because we have to move on to Marco's.
Yes, of course.
Great. Can you unshare your screen first?
Yeah.
Thank you. So we're now going to hear Marco Bigica's talk on selective impact of sleep deprivation on cognitive functions. And yes, take it away, Marco.
Cool. Can you see the full slide? Yes, nice. Right. Hi, everyone. I'm a PhD student at Cardiff University. And today I will give you a brief glimpse of the work I've been doing on the impact of sleep deprivation on the cognitive functions. So differently from the title, I will only focus on from the original title in the abstract. I will not talk about the brain in fMRI terms. But yes, hopefully it will be just as interesting, because yes, so there is really a lot of studies that have been done on the effects of sleep deprivation, and you may have known yourself from just not getting enough sleep how you feel after a night of poor sleep or sleep deprivation. But really what has emerged is that there are certain cognitive domains like vigilance and emotional processing that are really, really universally strongly impaired, and others like memory and selective attention and executive functions, which have also been studied, but have it has emerged that they are impaired, but maybe to a lesser extent or not in all individuals. And then there are also other domains like metacognition and perception, which are also, I think, quite interesting to study, but have received much, much less attention. So in my project, I'm really aiming to better understand how sleep deprivation affects a range of cognitive functions, and particularly these that have not received so much attention. So how do I do it? I've tested 14 participants during on this experiment that you can see on the slide. So basically, the experiment includes a 36-hour period of total sleep deprivation. That means one night without any sleep, and this occurs in the laboratory. So participants arrive in the morning around 7 a.m. and they will stay in the laboratory for about 36 hours. During this time, they get no sleep. We monitor their brain state with a headband, and at multiple sessions that you can see here on the top row, multiple times during the day, we assess cognitive functions using behavioral tasks, as well as we also assess their. We also take neuroimaging scans using MRI. And then they go home after 36 hours. They have a nice, comfortable sleep in their own bed and come back the next morning for a final recovery session. And as I said, I'm interested in a range of cognitive functions. So these are the ones that I'm looking at using different tasks. But specifically given the short time, I will just glimpse to three of these: vigilance, selective attention, and metacognition. So let's dig into the first topic. And so how does sleep deprivation affect vigilance? By vigilance, I really mean sustained attention. So our ability to remain focused for a prolonged period of time. To do this, we use a super simple task. Participants just fixate on a cross in the middle of the screen. When a counter appears, they have to press a button as fast as they can. So we count how many trials these participants have response times greater than half a second. So this counts as lapses. It's called a lapse. And what we observe, you can see on the x-axis the amount of hours awake. So each of these is one session. And on the left side, it's a baseline. So this is the first session they do in the morning. They have about 13 lapses on average. And then the amount of lapses that they do increases with the time spent awake. And then it goes back to baseline after a night of recovery sleep, as you can see from the right dot. So we can conclude that sleep deprivation does impair vigilance. This is well known already. Let's move into the more maybe controversial findings. For example, selective top-down attention. And we operationalized selective top-down attention using a visual search task. It's also a task that just required participants to fixate the cross. And there are a number of items that appear on the screen. One is a target, this T, and the other are just these similar distractors. Participants just have to press a button, a left, if depending on where the T is pointing. But we vary the set size, so the amount of distractions present on the screen. And what usually happens just normally is that the response time increases with increasing set size. And what we are particularly interested is this slope here. So the amount of increase between set sizes. What we expect if the sleep deprivation has a selective effect on top-down attention is an increase in the slope in sleep-deprived session compared to well-rested. However, we don't find a difference in the slope. As you can see here, these are in different colors, the different sessions. You can pretty much see, despite the overlap, that the slope is pretty much the same. And in fact, it's the interaction between the hours awake and set sizes is not significant, although there is a generalized increase in response time. So from this, we can conclude that sleep deprivation does not selectively affect top-down attention. So moving to the metacognitive part, this is particularly interested, I think, because metacognition is also our ability to think about our, for example, the decision that we made, the thinking about thinking, and so to measure this, what we use a perceptual task first. It's in this case, a grating is presented, it tilts to the right or to the left. Participants just say they move to the right or to the left, but then really the metacognitive task is that they have to rate the confidence about the accuracy in their judgment. So 1, if they are very unsure that they made the correct choice. 5, if they're really sure they made the correct choice, for example. And then we, using methods derived from signal detection theory, we calculate the metacognitive performance as the relationship between the confidence and the accuracy. And you can see this, if this shaded gray area is greater, then that means that they have better cognitive performance. If it's low, if it's small, it's poorer cognitive performance. And so our results here are normalized to the baseline, so relative to the session when they're just have just woken up and they're fresh. You can see a reduction in the area under the curve, so in a reduction in the metacognitive ability with the time spent awake. So when they're more sleep deprived, they have poorer metacognitive performance. And then I think this is particularly interesting because it really also relates to our assessment of our own fit when we are, for example, we didn't have a very good night of sleep and we want to drive. We think we are super fine, but our actually ability to judge ourselves is impaired. And so this may put us at further danger. And so just to conclude, I just summarize what we have found so far, that sleep deprivation affects cognitive function selectively. Why selectively? Because we see some cognitive functions that are strongly affected, like vigilance, others that are not particularly affected, like selective attention, and then some also that are also specifically impaired, like metacognition. And since we also acquired the neuroimaging scans in many of the sessions, we are now, I'm now analyzing these results to really try to understand why maybe different areas that underlie the cognitive processes involved in this task are more resilient, and hence they make certain cognitive function more resilient. Right. So thank you for your attention.
You're getting my applause here. Thank you. That was great. All right, so do we have any questions? I'm not seeing anything yet in the Q&A. Open questions. Okay, I will ask a question first. So you found this effect on metacognition in acute sleep deprivation. Like, if people are bad sleepers, would this effect be sustained like throughout the day?
So what we've done is an acute sleep deprivation experiment. So participants go without sleep for just one night. Findings about, say, sleep restriction. So sleeping less than what you maybe need to for several nights. There are no study that I know of about metacognition, but I can, for example, tell you that these prolonged effect of sleep restriction after a few days is just as bad as one full night of sleep deprivation with respect to, for example, lapses in vigilance and other cognitive functions. So I wouldn't know about metacognition, but it's possible. For example, subjective ratings of how sleepy you are have been shown to be significantly worse than actually objective rating of vigilance, as for example measured by this vigilance task. And this is an example of a metacognitive impairment. And I would have to look, but yeah, I may think that after a few days you are also impaired in our ability to judge yourself.
Okay, very interesting. Thank you. So there's a question in Q&A from Aldana. What impact do you expect in diffusion MRI?
Yeah, thanks for your question. So, for example, there are animal studies who have shown that the thickness of myelin is decreased after sleep deprivation. This may have to do with the function of sleep in reconstructing, repairing myelin, because during sleep there are lots of proteins and genes that activate that are related to this aspect. So with our diffusion data, for example, we expect to find a reduction in myelin thickness of axons.
Okay, I hope that answers the question. Apparently it does. Okay, thanks, Aldana. Cool. Do we have any more questions?
I could ask one. Marco, great talk. Your findings on metacognition, they remind me of basically when people drink. When people drink, I know that they aren't aware of how impaired they are. And so what my curiosity is how would I compare like the impairment of losing sleep versus being inebriated or something like that?
Yeah, I remember reading somewhere that they routinely make these associations, because many of the impairments that are observed after sleep deprivation resemble those that one can see after drinking too much. But yeah, you're asking how to do an experiment to compare the two.
Yeah, essentially, and it may be apples to oranges, but it does, like you said, it has a striking resemblance.
Yeah, I guess what may be interesting is if we find the same behavioral effects in a person who is sleep deprived and drunk, we may be interested to look at the neural underlying mechanisms. So whether they are the same or they are different, for example.
So you're going to have very interesting sessions at your scanner there, I guess.
Yes.
Is there any more questions there?
I have a short question.
Go ahead.
Yeah, so I was just wondering, like, it was very good presentation, nice presentation. So I was just wondering, like, when you're going to do resting state fMRI, as we all know that this resting state fMRI paradigm is a very sleep-inducing experimental design. So
yeah.
How to like confound your finding of this difference between?
Yeah, absolutely. That's a very good point. So our resting state is a 5 minutes resting state. We try to put it immediately at the beginning of the scan, so that we maximize the ability that they are awake. We have no eye tracker, so we can't track them. So that's a limitation of our study. Immediately following the sleep deprivation, immediately following the resting state scan, there is in the same bold sequence, after 5 minutes resting state, it starts another 5 minutes of the same psychomotor vigilance task that I showed you earlier, just in the scanner. So one way that we have to sort of see if they are awake is that they are, if they immediately respond to the stimulus, then we know that they are awake. If they don't respond, in 1 or 2 instances, for example, they fall asleep, so we had to redo the scanner because we knew that objectively they had fallen asleep. Yeah.
Thank you very much.
Yeah, no problem.
Sounds good. Very interesting. Okay, let me see. I don't think there are any more questions. So I want to thank all the speakers again. Applaud on everybody's behalf. This was great. I learned a lot. I hope everybody else had a good time too. And yeah, thank you for coming. Thank you for your presentations, and enjoy the rest of NeuroMatch 3.
Thank you, Marius. Thank you, everyone. Bye-bye.
Bye-bye.
Identify the cells, and you know, at some regions in the image, identifying the cells is going to be a bit harder. So we should kind of like, you know, incorporate information both from the spatial posture of the worm as well as the colors of the neurons to determine where the neurons are. And then once we determine the identity of the neurons, then we also have like videos and you know, together with this biological technique, which is called NeuroPal, we can also express GCamp in the worms, and then can record the activity of the neurons as the worms are moving and behaving. So what we did was to grab these data sets and develop some pre-processing pipelines to extract information from these videos. Namely, we actually developed multiple methods that I'm listing here. So the first one is atlas construction. So we know that the worm body and worm neurons are kind of a stereotypical. So we can go ahead and you know, use this as stereotypical stereotypy to kind of develop a statistical atlas. So this is how the atlas looks like. So we are assuming that there is a latent space where every neuron has a center and it has like some sort of covariance matrix. And then different worms are actually draws from this distribution. So we can try to incorporate all the information from multiple worms to develop a statistical atlas of neuronal positions and colors. And then what we can do is that we can, for a new image, we can try to register this new image to the atlas that we have developed. So this is what we did. So there is another work where we can grab this atlas, which is this a red point cloud, and grab the neurons in the image, which is this green point cloud, and try to match these neurons, align these neurons together. Once we do this, we have the aligned space, but then we have to determine, based on the neurons in the atlas, we have to assign an identity to every neuron in this image. So for that, we used a method that is called a Sinkhorn's algorithm. So it, you know, gives us some sort of probabilistic assignment of the neuron. So we can say that neuron that is appeared in a certain position is 50%, has 50% probability to be assigned to neuron A and 50% to neuron B. So this way we can also deal with uncertainty. We furthermore developed a probabilistic segmentation method where starting from this statistical atlas, we can actually write down a Gaussian mixture model with some sort of prior distribution and treat the pixels in the image as Gaussian observations, and we can fit this model and you know, get the segmentation in an unsupervised manner without having training data. And finally, we also did some work in demixing calcium signals in the moving animals. So when the animals are moving, we cannot use traditional matrix factorization models because they're assuming that the tissue is fixed. Since the tissue is moving, we introduced this new like functional form, which is we are transforming this canonical space into different time points. So there's some sort of deformation in this model. And yeah, these are basically the multitude of the methods that we have developed for analyzing this data. And many of these methods are also applicable to other model organisms. Finally, I'm listing the papers that we actually published from these methods, and some of them, 3 of them recently published in MICAI. And I also wanted to note that the code and software are also all available on my GitHub page.
Thank you, Armin, for the nice and brief descriptions of your work. If you have any questions for Amin, please post them in the questions and panel sessions or through the chat, and we will read them for him. Amin, could you please explore, is it possible to use this method for other animal models? And
yes.
If so, what kind of modifications or skills do we need to be able to use it for other models?
Yeah, that's a very good question. So yeah, for sure, many of the models that we developed, they can be applied to other model organisms. In order to develop an atlas for a different model organism, we need to have some sort of a stereotypical aspect of the data. So here we know that the neurons relative to body are appeared in the same position. So if you have that, that's sort of, and this is very much similar to what is done in other data sets, like MRI, for example, where you actually develop some sort of atlas for the brain, for the human brain, which is like an average human brain. So this is what we're actually doing here. So all these steps that we are following here are very similar to what is done for human MRI. Atlas construction, image registration, detection of different regions, and then segmentation, and then like reading fMRI signals, for example. So this is very much in the same spirit. Yeah, so like the atlas depends on the the stereotypical and this other method, which is just the point cloud registration. There's like a rich literature on that. We also did some theoretical work. So if you have like 2 point sets, you want to register them, that depending on the type of problem, you can definitely do that. If you want, like if you have 2 images, you want to find some sort of assignment, or identity detection between these 2 images. You have like 2 point clouds, you want to match them. The segmentation, although we are using this information about the atlas, but this assumption can be relaxed, and then we can try to just go ahead and without a prior information, just do the segmentation in the image without detecting what the identity of the neurons are. And this is just a general segmentation method, and we showed that it works better than many of the existing segmentation methods. So this is also applicable to other model organisms. And finally, signal demixing. So I think there is a lot of one of the limiting factors of current signal demixing approaches is that they cannot deal with the motion. So you need a method to actually fix the tissue before and do some sort of motion correction before running the matrix factorization models on for demixing calcium signal. But here we are relaxing that assumption, and we are assuming that there is some sort of deformation, that we are capturing the parameters of the deformation at the same time that we are actually reading out the signals. So yes, this is very much applicable to other model organisms depending on the type of problem.
That sounds really good. Do you have any support or documentation to use it to modify these softwares, or are you planning to, for general use?
Yeah, I think what we have been trying to do was to kind of develop very clean and very structured codes, and for many of the applications, the codes are like very structured. So I don't know, like maybe I can just give you an example of just go through my GitHub page. So this is we have been using like object-oriented programming, so the codes are very structured. There's a demo file for each of the codes that there's simulation, so you simulate data and then you go ahead and run your segmentation. But then like there are different ways for calling the function of the segmentation. You can actually relax all the assumptions. You can remove the prior from that. So yeah, like there is some documentation in the codes that are on my website. But if anyone is interested in a specific use of these methods, then they can definitely reach out to me, and I'll be more than happy to help with different helping with different applications of these methods.
Yeah, that sounds good. Thank you very much.
Thank you.
And if you have any more questions, please reach out to Amin through his Twitters, which shared in the chat, or he will be around here throughout the sessions. Please feel free to post your questions. He will be happy to answer for the details and other concerns. Now we will move to our next speaker. And thank you again, Amin. It is, I really enjoy this kind of open softwares because they make the neuroscience more applicable to general use, especially for this kind of complex data sets.
Thank you.
And for our second speaker now is Jesse Lauer. He got his PhD from University of Porto, Portugal in 2017, during which he studied the mechanics of human movements performed in the water. Then he worked 2 years in a project to use biomedical modeling to inform rehabilitations of motor impairments. This year, he joins McKinsey and Alexander Mattis Lab as Deep LabCut's next core developer. His talk is titled Multi-Animal Pose Estimation with Deep LabCut. Stage is yours, Jesse.
Thank you very much. Let me just... Can you see my screen?
I can see you, but not your screen.
Let me see that again. All right, yeah, it should work. Is that okay?
Will be in a minute because it's taking some time to search it. Yeah, we can see it now.
Awesome. So yeah, thank you very much to you for the very kind introduction, as well as people who tuned in. So I think we can all agree that deep learning has revolutionized the way that we measure animal behavior in a non-invasive manner. But looking at those videos, I think it should be clear to everyone that estimating their poses, as well as tracking those animals across time, is a real challenge. And so today, I'd like to illustrate what the challenges are and take you through the solution that we found using our toolbox, Deep LabCut. So since we can't possibly solve that problem all at once, starting from a raw video to the final output, we decided to break it down into more tractable sub-problems. And so the way that we do this is our approach is bottom-up. That is, we decided to build onto the strong Deep LabCut detector for body parts. And then we build up to assemble those body parts together into animals. Then we need to track those animals across time. And once we obtain tracklets, then the final goal or the final sub-problem is to stitch those tracklets together in order to form the final output. So our solution to the first sub-problem, animal assembly, is iterative. That is, we will connect body parts together by going, by looping from bone to bone in the skeleton. And then we connect those body parts that obey certain roles and parameters. And 3 of those parameters are absolutely essential. So this is the 3 parameters you can see here, path threshold, disnormalization, and detection threshold square. And those, like I said, they are essential, and they are validated, say, by Bayesian optimization. Then we moved on to investigate the effect of using redundant skeletons on the accuracy or on the quality of the assembly. And what we realized is that using redundant skeletons, but what I mean by redundant is using a skeleton that has redundant connections between body parts, has a great beneficial effect on not only on the number of unconnected body parts, that is, we reduce by a factor of 4 the number of body parts that are not connected, but we also drastically improve the overall quality of the body parts that are correctly assigned the right animal. And once those animals are assembled, then we are left with the second sub-problem, which is how we will propagate the identities of those animals across time. And the way we do this is we adapted a lightweight framework where for every given frame, we will try to match the detections, so the animals in the scene, with predictions from trackers that are taken from the preceding frame. So in other words, what we're trying to do here is to find one-to-one correspondence between adjacent frames. And this is the process that we repeat every single frames in order to propagate identities forward. And what we see is that, especially using our newest animal trackers, that tracking accuracy is near perfect, or I'd say excellent. But we're still left with the problem of fixing identities switches or swaps, which very often occur when animals get occluded or pass very close to one another. So what we do here is we recognize the need for a global solution to that final problem. So once we arrive at that final stage, you are left with a set of tracklets that you see here on the left of the screen. And the goal will be to find an optimal way to stitch them together such that they are kinematically consistent. And the efficient way that we found to solve that problem, which is way different from the way we currently do it, is to cast the problem as a network flow optimization problem. So I won't get into the detail, but the idea here is if we can find flows through that graph such that the cost of that flow is minimum, then we immediately obtain a globally optimal track. And what we see, for example, is that using that method, we reduce by a factor, by at least a factor of 2 up to a factor of 7, the number of identity switches. So just to wrap up, we know and we know that pose estimation, whether it's single or multi-animal, is very robust, which is why we decided to build onto this. But then in order to track multiple animals, we need additional steps, which is to assemble those animals or detections into animals. Then we track those animals through times. And in the final stage, we stitch those tracklets together. And I just like to remind people that the code is open source on GitHub. It's still in beta, but the official version will be released quite soon. It's like quite some nice additions. And a big thank you to all my lab mates, the PIs, the contributors and users of DeepLabCut. DeepLabCut was originally published like 2 years ago, and we've recently published a primer in here, and that is, I believe, in free access. So I'd encourage everyone that is curious to understand more about pose estimation to just go and have a look. And thanks to CZI for funding me.
Thank you, Jesse. It is very, I'm also using the DeepLabCut for pupil analysis. It is very useful and really easy. It has good documentation and everything. But yeah, one question while we are, please ask your questions in the question and answer panel for Jesse, DeepLabCut software. One question I have for you is like, currently it is in the Python. Is there any direction for using a MATLAB version of it or creating a MATLAB version of it?
Wow, that's a hard one, because the core of the package, right, is the whole machine learning architecture relies very heavily on Python, packages that are written in Python. So I don't know. That's a tricky question, because I have no real idea about what it will take to actually even build bindings to or bridge, let's say, Python and MATLAB. But my feel is that obviously that's something I'd be considering, but I also feel that we've been spending a lot of energy into having notebooks that are pretty much ready to run, where you just execute pretty much every cell. And so I think that the barrier anyway to use it was quite low, but MATLAB would be a big piece.
Hard one. Yeah, I think for many in the academia. Yeah, thank you. We have a couple of questions for you. First one is from Edmund Telly, and Edmund is saying that he's guessing tracking marmoset is more difficult than mice. Can you use animal-specific physical markers to facilitate?
Excellent question. There is something indeed that I do not mention during that short talk, which is that's something that we've been implementing quite recently. And actually what it does is that then our network would also predict the identity of every single body part by precisely learning from some physical features of the animal. And in the marmoset case, for example, like, I don't think that was very clear from the video, but one of them had some blue dye on one ear to make it, slightly recognizable from the other. And we could, for example, use that feature to aid, I would say, in the process of recognizing what animal is which animal is which one, and also aid in the process of stitching the trajectories together. That would be an extra information to give to the entire pipeline to facilitate or to make the whole process more accurate.
Thank you. We have another question for you from Joseph Lutsinger. Joseph says, great talk, Jesse. How does time of animal overlap change how hard it is to keep each animal correctly identified?
Thank you for the question. So, excuse me. So the thing is, this is probably something that is hard for the second subproblem that I mentioned. So if animals interact or get close to one another very often, then this is likely an issue for purely tracking and propagating identities on a frame by frame. But the good thing is, we very recently understood that for the last subproblem to solve, so stitching tracklets and doing it differently than we currently do, that is, the way we currently do it is we basically solve the problem very locally, and this is the way we stitch tracklets together. And so shifting from that perspective and adopting an approach that is more global, we realized that we ended up with tracklets or tracks that were of much higher quality, such that this would correct precisely identity swaps, and this would work very well for even challenging data sets. So.
Okay, thank you. And
I'm sorry, I can't see the question, so I'm not 100% sure that I answered to it fully. But.
And yeah, there is this question and answer panel down. If you click it, you will be able to see the questions as well on the screen next to the share screen button. And the other questions is from Christian Ebesan. Christian says, it's very cool. It went a little fast. Could you perhaps show the slide with the accuracy in the mice after tracklet stitching again? In an earlier slide, it looked like you had 0 switches in the mice, but some switches with the tracklet switching.
All right, so I guess this is the slide that that is being referred to. So here indeed, oh, excuse me. Yeah, indeed, the multi-mouse data set is actually very simple to solve, and using our 3-stage, let's say, approach to the multi-animal problem, then we could just stop here and we would have amazing reconstruction. And I completely here, I just discarded the multi-mouse data set. So I'm only talking about the reduction in the number of switches that we observed on the 2 most challenging data sets that I introduced. So naturally, since there was no switches before with the multi-mouse, and we end up with tracklets that are extremely long, there is no way we would introduce switches by just stitching them together, since they are essentially already stitched.
Okay, thank you. And Joseph, for the previous questions, he says, that was great, but he has another question for you. He says, do you expect tracklets will improve ethograms, behavioral identification whenever that is implemented?
Yeah, definitely, right. In the sense that if we, from the tracking phase to obtaining the final tracks, if we end up with tracks that are very pure in the sense that they pretty much require no user input in order to correct for those swaps, then I guess this is very high quality data for any behavioral analysis or anything coming afterwards, right? I think those are just techniques to make users and also us developers more confident that whatever enters in a post-processing pipeline is of very high quality. So I would definitely believe that this is yeah, this is extremely beneficial to whatever analysis is coming next.
Okay, thank you. For Christian, for the previous questions, he says, you know, you mentioned 35 switches per what? Is it like in an hour? Or, you know,
okay, so this was
switches?
overall across the entire video. So the fish actually, I could even show it again, but the fish video, yeah, the fish video had 600 frames, but it's extremely challenging. So that is across all those interactions that you see here, and this is, I didn't count it, but that's probably 200 possible crossing and risk of identity swaps. Then we took it down to only 35. And for the marmoset case, I think this is 15,000 frames also, where they are also interacting together and hiding and, well, you know, passing close to one another. And we took it down to, yeah, from 200 or so identity swaps to 30 something. So this is, yeah, that's an absolute number of switches, of identity switches.
And thank you. I mean, you have less than a minute, but I'm just going to read the last questions from Chanvigo. For a new user to develop code, where should we start? Is there any platform for learning?
So I think the best way would be first to go to our GitHub repo, because then we have amazing documentations, we have great tutorials, access to, well, and I see that Mackenzie is kindly giving the link. So, well, anyway, we have a lot of resources, whether it is videos and Jupyter notebooks to get started. And also if he faces any issues, not to hesitate to meet us on that forum, I guess. I think you have sent the link earlier, where you should feel free to just ask questions, and the whole community would be more than happy to answer.
For me, I was using the GitHub, the tutorials folder you have. It is quite useful to start with. Yeah, Matis just shared the develop code for you, which I'm going to share in the chat panel as well for you. And yeah, feel free to reach out. Okay, so thank you very much, Jesse. This is really amazing. And now we are going to move on to our next speaker. Our next speaker is Sarah. Sarah Stetnitz. She is a postdoctoral fellow in the Zopping Lee Lab at the Max Planck Institute for Biological Cybernetics in Germany. Her talk is titled "Zebrafish Exhibit Visual Attentional Pop-out Effects in Their Behavior." Stage is yours, Sarah.
Thank you for the kind introduction. So I'm in a lab that primarily is focused, historically has been focused on questions of visual attention. I'm also co-advised by Jennifer Lee and Drew Robson, who do a lot of really fantastic microscopy and behavior experiments, specifically in zebrafish. And I hope that I can sell you on the end of this talk for the utility of studying this behavior that's common essentially to all vertebrate species in an animal that is the size of an eyelash. So what is the problem of attentional selection? And animals have a problem with their sensory environment, right? This is exemplified by vision, especially, where there's much more information in a visual scene than could ever be processed by the brain. So if you're not going to spend 30 minutes processing every single scene that you look at, which of course is not advantageous to your survival, you need to have strategies for directing your attention only to parts of the visual scene that are important. So one of the hallmarks of this kind of bottom up, so very rapid attentional selection, is called the pop-out effect. If I show you 10 dots and one of them is a unique color, or I show you 10 bars and one of them is a unique orientation, they immediately draw your attention before you even have conscious awareness of it. So what does that actually look like as a behavioral readout? We call it orienting. So it takes many different forms. In humans, you might have a gaze shift. For example, when I switched to that slide with the single red dot, you probably looked at the dot before I even began speaking about anything. Other animals might move their body parts, like a cat could rotate its ear towards something that it heard in the environment. But in our model system, the zebrafish larvae, they actually will do an orienting turn and move their entire body. But regardless of the mechanism for directing the sensory organ, eyes or ears or body, towards this relevant stimulus in the environment, it's essentially the same type of behavioral readout. It shows you that an animal is selecting and directing its attention towards something in the environment. So why are we using the zebrafish specifically? Like, this is certainly an established model organism in genetics, but it's also a really great model for neuroscience, and visual neuroscience in particular. They have many behaviors that are entirely dependent on vision, that occur very early in development. They have evolutionarily conserved brain regions that are devoted to visual processing. So the optic tectum is functionally equivalent to the superior colliculus in mammals. And of course, my favorite part about them is that they're really amenable to this fabulous live imaging of neural activity. So we can do, on the single cell scale, whole brain recordings in a freely behaving animal, which is simply not possible in other vertebrate systems. So the experimental setup at this stage is behavioral, right? We want to study the underlying computations that permit this behavior to happen. We need to demonstrate that it actually occurs in our animal the size of an eyelash. So we have our fish in an arena, and we're recording from below, doing live tracking of the body and the head position. And we're using that to, in real time, generate a stimulus and send it to a projector. We then project stimuli above the animal's head in different positions, colors, combinations relative to the body axis and the position of the eyes. So to give you an example, for these experiments, we're using fictive prey. So a zebrafish larva will hunt these little single-celled organisms, paramecia, and we're giving them something that mimics that. And we can place these fictive prey at different positions along the midline of the fish. And by measuring their orienting turns of their body after stimulus onset, we can get a direct readout of what they're paying attention to in their visual environment. So the first test was to make sure that we really could get these animals to chase light. So we ran these experiments using just single white dots placed at 8 possible locations in the visual field. So these are placed on the left side and these dots are placed on the right side. And if you look at the change in heading, so the orienting body turns within 1 second after stimulus onset, when you present a stimulus to the left, we can get them to turn to the left. When you present it to the right, you can bias them to turn towards the right. So they are paying attention to these artificial prey, even though they can't interact with them physically. And this is occurring at quite a low latency. It's looking like 200 milliseconds, which is more or less what we would expect with this kind of very rapid attention selection. And we can pull out all kinds of nice extra pieces of data from this. Of course, we want to know what the optimal position is to evoke an orienting response. And it turns out that it's about 45 degrees from the midline. This is nice because it agrees with naturalistic behavioral analyses of prey capture that other groups have done. So sanity check is always a good deal. But we also ran these same experiments using different colors. So this is going to be necessary for the subsequent visual attention experiments. We need to know, do they respond to red, green, and blue? The answer is yes. Regardless of whether you present it on the left side or the right side, both red, green, and blue dots can evoke the same type of orienting response. So why is the color important, right? I like to use the color pop-out example because it's the most immediately obvious, I think, to people and likely to fish. Fish have excellent color vision. They see red, green, blue, and ultraviolet. So instead of just showing 1 dot now, we're showing 8. So I've provided a stimulus that is 7 distractors of a uniform background color and a single pop out singleton. So that's one dot on either the right or the left side that's unique from all the other dots around it, although there's the same number on either side. And what we're very excited about is that we were able to bias motion towards the side that the unique stimulus is placed on. And that's regardless of whether it's a red singleton with green distractors or a green singleton with red distractors. And so the next step for us, now that we've demonstrated that this tiny little brain is making this kind of computation, is to take this onto the microscope, where we can actually look at the activity of single neurons and how they're interacting with each other to directly test some of the computational hypotheses of how the pop-out effect is implemented both in humans and in fish. So with that, I'd love to take questions either about the system or the experiments.
Thank you, Sarah. This is so lovely. I think it must be really nice to work with fishes. They are, I believe, cute. We have a couple of questions for you. Elena Jacobs, first question is from Elena Jacobs. Elena is saying, really cool work. And I believe the fish are freely moving. How do you control where in their visual field in this the stimuli occur? Or do you determine this post-hoc?
So we are doing live tracking. This is definitely one of the biggest challenges of implementing this kind of system. We really want to make sure that what we're showing the fish reflects what the fish is actually seeing, right? So it requires this on-the-fly tracking. At this stage, we're doing it at 60 frames per second, and we basically only need an instantaneous snapshot. So we know where the fish is when the stimulus comes on, and then it stays in the same position. So it doesn't actually update its position with the fish, which helps in case of tracking errors and things like that. But it certainly has been a lot of technical innovation and calibration to make sure that this is really robust and automated.
Okay, thank you very much. For the other questions, unfortunately, I don't see the name clearly for the person who is asking the question. It's like mmazer.17@ucl.ac.uk. He says, this is very, very cool. In explicit report paradigms in humans, a pop-out effect is also observed for color absence and nothing that no target is present. Is this something that has been or can be tested on larvae?
Yeah, so there's no reason we can't do all of the same experiments we do in people and in fish with this setup, which is one of the best parts about it. I think our one limitation is the number of distractors that we can present, because there's only so far away that we can, they can see, right? I think that's an awesome idea. That would be a great experiment to implement. I can tell you the other piece of data I have from the pop-out experiments that isn't shown in this talk is uniform. So 8 dots on all sides and there's no singleton. And that you get stochastic turning. So it would be fabulous to see that kind of symmetry breaking just by the absence. That's a great suggestion.
And for another questions, thank you. For the previous questions, the name was Mayton Mazer. Thank you for clarifying your name, Mayton. And for the next question, from Inbal Shaner, he says, very cool. Are the 8 dots are constantly presented? Is the singleton surprisingly pops out?
So the 8 dots are constantly presented. And you bring up actually an interesting point, because motion pop out is something that has been studied in different species of fish. There's a species of fish called the archer fish that actually sprays water at its prey. And they've been able to train these animals to target different pop-outs. So this is not a system that's amenable to live imaging, of course, which is why we're trying to do it in zebrafish. So for these experiments, the color pop-out, they all appear simultaneously, including the singleton. The reason for that is if you change your stimulus, that could then become a motion pop out. We're really interested in doing those experiments. And I think You know, it'd be cool to do orientation. It's a little challenging because of the projector system, you're projecting from above, but I think it's quite likely that the same kind of effects we see with this color paradigm we would see with motion, like a change in the stimulus.
That sounds so cool. Thank you. And for the last questions from Ameya Menon, says so cool. How could you please go over once again what kind of bias you observed when presenting multiple colors?
Yeah, so in the case of like the human pop out effect, right? If I just showed you these 8 dots and one of them is unique, your attention is going to go towards that unique dot. Because we're using these orienting turns of the fish as our readout, we would expect if the pop out effect is working in our system, that if we present something that's unique on the right side, the animal would make an orienting turn towards the right side. And that is what we see. And I think very critically, one of the most important things about these data is that both combinations work. So I've gotten a lot of questions about whether or not there's innate color preferences. Like if they just like red dots more, then they're always going to turn to the red dot, right? And perceptually, the green channel on our projector is actually probably quite a lot brighter. So maybe it's just the sheer number of photons that the fish is getting on its retina, right? But the fact that in both cases, whether you have red singleton and then 7 green distractors, or a green singleton, 7 red distractors, we can bias them to turn only towards where the unique stimulus is. I hope that clarifies.
Thank you very much. And if you have more questions for Sarah, Jesse, or Amin, please reach them out through Twitter or the email which is shared throughout the chat. And thank you very much, Sarah, Jesse, and Amin. It was very lovely to hear your work. And thank you very much for our audience who was asking very interactive questions. And I also would like to thank to Melvin for giving us technical help at backend. He was here throughout the session, which was helping us for the technical issues. I hope you enjoyed these sessions and will enjoy the rest of the Neuromatch conference and following sessions. Thank you very much, everybody.
Thank you.
So we are on YouTube as well, or will be.
Okay, let's start. So hi everyone, thanks for joining us. We have a great lineup of speakers. We'll start with a cognitive talk by Sricharan Sundar. Sricharan holds a bachelor and master's in metallurgical engineering before pursuing a PhD in the cognitive sciences in the Indian Institute of Science in Bangalore. The title of his talk is Posner cueing specifically modulates attention, but not expectation, behavioral and neural evidence.
All right. So can you hear me fine? Right. So thanks, Shlomit, for the nice introduction. I am Sricharan Sundar, and I'm going to talk about my work with Kavya and Dr. Sridhar at the Cognition Lab at Indian Institute of Science. My talk is titled Posner cueing specifically modulates attention but not expectation. Right. So what are attention and expectation? So attention is the process of filtering behaviorally relevant information and filtering out the irrelevant information. Whereas expectation is the process of using prior information. For example, here, you might perceive this as a ship, but in reality, it's just a bunch of meaningless pixels, right? So thus, attention can, both of these processes can modulate either sensory processing, decision-making, or both of them. Now, we quantify the modulations to these by a theoretical framework called signal detection theory. And it indexes the sensory processing by a parameter called sensitivity, which we call D prime, and the decision making by a parameter called bias, C, right? We are going to talk about these processes in the context of a Posner queuing task. Now, what is a Posner queuing task, right? It's a very widely used task in attention literature, and it proceeds as follows. A fixation cross comes onto the screen, and then two stimuli come up on the two sides, and then a cue can point to the right or the left location, right? Now, after a little while, one of these stimuli changes or none of them change, and the subject has to respond which one, the right or the left one changed, or by a third response if none of them changed. Now, the cue points to the stimulus that is most likely to change. Now, since the subject has to also localize this change, the cued location becomes behaviorally relevant as well. Thus, they also attend to the cued location. So as you can see, then the attended location, which is the behaviorally relevant location, is same as the location which with high stimulus probability, that is the high probability of change. Now, this begs the question, are modulations in Posner queuing then because of attention or because of expectation, right? One way to study this is to decouple these processes in behavior, and then to ask which one does the Posner queuing modulations correlate with? Does it correlate with attention or expectation modulations? So fortunately for us, a previous study by Wyatt et al. Designed a paradigm to decouple these processes in behavior. And what they observed was that attention modulated only sensitivity and expectation modulated only bias, right? Now the question we asked was, is the Posner queuing an attention cue or an expectation cue? So we looked at it from two aspects. One is behavior and the other is EEG. So now what was this task by Wyatt et al., right? So it's called a response probe task, and inspired by that, we designed a similar task. Our task proceeds as follows. First, there's a fixation cross that comes on the screen, right? And then the color of the fixation cross tells you which side has higher probability of change. After this, two stimuli come up, and 350 milliseconds after this, an attentional cue comes up. This lasts for an exponential period of time, right? And during this exponential period of time, the stimulus doesn't stay on. It stays on for 250 milliseconds and goes off unpredictably between 50 and 300 milliseconds. After this, there's a blank, and then a change comes on. So the change could have happened in both stimuli, one of the stimuli or none of the stimuli, right? The subject then is probed for a response. The subject has to respond whether the stimulus at the probed location, which is here, the left location, changed or not. Now, the attention cue, which is this, it tells you most likely to be probed for a response, right? It doesn't give you any information regarding where the change is probable to happen, right? So thus here the attention and expectation cues, both of them are statistically independent, and we ensured that that was the case. Now the subjects who did this task also did a Posner queuing task. And as I explained before, the Posner cue points to the location of higher probability of change, and thus the location of higher behavioral relevance, right? The subject had to give one of three responses. Did the left stimulus change, right stimulus change, or none of them changed? Okay, now let's look at what modulations these behavioral modifications give us, right? So here you have three columns. The first two are from the response probe task. The first column is because of the modulations by attentional cueing, second by expectation cueing, and the third column by Posner queuing. The first row is for sensitivity, and the second row is for bias results. So what we observed with attentional cueing is that it modulated sensitivity, but did not modulate bias, whereas expectation cueing did not modulate sensitivity, but modulated bias, right? Thus, we replicated Wyatt et al.'s results. Now, Posner queuing, on the other hand, modulated both these parameters, right? It modulated sensitivity as well as modulated bias. So to summarize, attentional queuing modulates sensitivity but not bias, expectation queuing modulated bias but not sensitivity, and Posner queuing modulated both. We then investigated which of these modulations did Posner queuing correlate with, right? To investigate this, we first asked whether the Posner queuing modulations correlated with attentional queuing modulations. And for this, we looked at we asked whether sensitivity modulations by Posner queuing was correlated with attentional queuing modulations in sensitivity. And if indeed Posner queuing is an attentional cue, right, these two measures should be correlated. And we did find that that's the case across 19 subjects. Now, then we looked at whether Posner queuing is also acting as an expectation cue. For that, we looked at whether the modulations in bias by Posner queuing correlated with modulations in bias by expectation queuing. And this was not the case, right? So behavioral modulations by Posner queuing correlated selectively with sensitivity modulations by attentional queuing. Now, we also looked at the neural signatures of these processes. And for that, we looked at three measures, right? Steady state visually evoked potentials, which have been implicated in attention, alpha oscillations, which have been implicated in attention as well, and beta oscillations, which have been recently implicated in expectation modulations. And we asked the same question, right? Did the Posner queuing modulations correlate with attentional modulations or expectation modulations? First, let's look at SSVPs. On the y-axis, you have power at SSVP frequency, and on the x-axis, you have time from fixation onset. The solid line is the stimulus onset, and the dotted vertical line is the cue onset. Now, with attentional queuing, what we observed is that, yes, attentional queuing did significantly modulate SSVP power, and that can be seen from this bar plot on the bottom left, that they were indeed significantly modulated. Expectation queuing, on the other hand, did not modulate SSVPs at all, but Posner queuing, on the other hand, did modulate SSVP power, which suggesting that effects of Posner queuing on SSVP power matched those by attentional queuing. Now, what do we see with alpha power, right? We plot the same plot. Y-axis is alpha power, x-axis is time from fixation onset, and the vertical lines are as before. So with attention, what we observed was that attention modulated alpha power post-Q, and that was significant, as can be seen from the bar plot on the bottom left. Expectation did not modulate alpha power post-Q. Posner queuing, on the other hand, did modulate alpha power, right? So again, from this, we see that effects on Posner queuing on alpha power matched those by attentional queuing and not expectation queuing. Now, the third thing we looked at was beta band power, right? And beta band power is known to modulate with expectation, right? First of all, we asked whether beta band power did indeed modulate with expectation. And we observed that it did indeed significantly modulate with expectation, right? Now, the question is, if Posner queuing was indeed an expectation cue, we should observe beta band modulation in Posner queuing as well. But what we observed was a null result, right? Attentional queuing and Posner queuing both did not modulate beta band power, right? This suggests that, so to summarize, beta band power was modulated by expectation, but not by attentional or Posner queuing. And to summarize my talk, behavioral modulations by Posner queuing correlated selectively with sensitivity modulations by attentional queuing, but not by expectation queuing. Posner queuing modulations in EEG signatures also correlated with attentional and not expectation queuing modulations, right? So both of these lines of investigation seem to suggest that Posner queuing is indeed functioning as an attention cue, but not as an expectation cue. And that's what we posit. Thank you. And I would be ready to get any emails from you guys regarding this talk, or you can ask me interesting questions here itself. Thank you.
Thank you very much for your talk. It's great to see how old questions still getting attention and novel results. We have one question here by Brandon Chen. I'll read it. Did you manipulate expectation at the block level, as in the wired task? Does this influence the interpretation of the Posner cue as more like an attentional cue? That was the question.
So we did indeed modulate expectation as blocks, because that is important for the subjects to develop it as an expectation implicitly. But what we did was that the motor response that the subject had was not fixed. So the button he had to respond to for change and no change, they were randomly assigned at the end, that the subject got to know only at the end of the trial. Thus, there couldn't be any sort of a motor bias that is contributing to these effects. And to control also, for the blocking that we did in the response probe task, we also blocked the Posner queuing task to control for those modifications that we did to the response probe task. So we do think that Posner queuing is not really an expectation queue, but an attention queue, because we did all these controls. I can't hear you.
Sorry, I was muted. We have 2 minutes for more questions. In the meantime, I'll ask you, I think I saw in one of the slides, CNV, or anticipatory activity. You didn't talk about this, and if you could explain this a little more, the CNV.
That anticipatory activity before the stimulus onset?
Right after. Yeah.
In which result was this?
I think I saw it in 2 conditions that they were separated. But never mind these slides. Did you observe any CNV effect?
So we did not observe. I'm not aware of the CNV effect that you're talking about, but then we did not observe any anticipatory effects as such, because that would show up sort of before the attentional cue comes up, right? If there's any sort of an anticipation. Also, the second thing is that the subjects, the event for the relevant event is the change onset, right? And the subjects didn't know when to expect that change. So because of the exponentially distributed cue, they had no idea when to expect that change. So I don't know if we will even see any anticipatory activity. Over and above that, the stimulus itself was switching on and off, right? So that we did so that the subjects sustain their attention, but then that also has the effect that there can't be any anticipatory activities because of that manipulation. I mean, it would be great to know what you think about that.
Okay, that makes sense. All right, thank you very much again, Switzerland, for your great talk. And we're moving on to the next speaker, Ksenia Kozlova.
Thank you.
Ksenia Kozlova is working at the Neurodynamic Research Group at the Institute of Cognitive Neuroscience of Higher School of Economics in Moscow. And her title is PMA as a model to study the somatotopy of the interhemispheric interaction.
Can you hear me? Excuse me, can you hear me? No?
Yeah, I can hear you.
We can hear you, Ksenia.
Okay. All right. So, as you said, I work in a high school of economics and neurodynamic group, and today I want to present you physiological neuroactivity as a model to study the somatotopy of the interhemispheric interaction. With regards to the background, balanced interhemispheric interactions are important for coordinated movement execution. The violation of interhemispheric interactions can be observed in patients after stroke or in patients with movement disorders such as vascular Parkinsonism, dystonia, tremor, or congenital mirror movements. It's involuntary motor activity of the contralateral limb. For motor rehabilitation, approaches dedicated to balancing interhemispheric interactions are commonly used, such as non-invasive brain stimulation methods, TMS and TTCS. But the selection of the methods of influence, facilitation, or inhibition of the unaffected hemisphere is a complex topic. And the reason is in the ambiguity of the question of individual assessment of the interhemispheric balance, the question that our research is focused on. For today, there are three models of interhemispheric interactions. One of them is the curation model that tells us that contralesional hemisphere is inhibited by ipsilesional hemisphere. Second one is about that contralesional hemisphere compensate functions of ipsilesional hemisphere. And at the combination of these two models, there is a new bimodal model, which assumes that types of effect from unaffected contralesional hemisphere depends on the structural reserve. And different type of interhemispheric interactions may be either beneficial or detrimental in the different subjects. To study this, we need to have reliable method of interhemispheric interactions assessment. There are different methods to do it, but analysis of features of physiological mirror activity is one of them. And we assume that physiological mirror activity may help us in the assessment of personal interhemispheric balance. We study features of mirror activity such as duration, latency, persistence, and amplitude, while somatotopic parameters of mirror activity may tell us about selectivity of interhemispheric interactions. Stepping back, physiological mirror activity is an involuntary muscle activity in one limb muscle that happens when muscles of the opposite limb are strongly contracted. Like on this picture, you can see pinch test and the recording from FDI muscle on the right hand, left hand. And while performing this metal test, we can observe this activation of muscle contraction and involuntary activity on the left hand in the humongous muscle. Until now, the study of physiological mirror activity was carried out primarily in the homologous muscles of the opposite limb, and the question about muscle specificity of the phenomenon is not clear. We hypothesized that physiological mirror activity may serve as a model to study the somatotopy of the interhemispheric interaction, and while performing isometric contraction of different upper limb muscles, mirror activity will be more prominent in the homologous muscles of the opposite hand compared to other muscles. Like here, the ADM during little finger abduction, we are hypothesized that PMA is more prominent on the same homologous muscle compared to the to others. In addition, this question is relevant for brain-computer interface. A motor imagery-based BSI relies heavily on the modulation of endogenous neural oscillation, which reflect cortical excitability. Since effective BSI requires quick-up and down-regulation of the excitability in the left hand and right sensomotor areas, this research may answer to what extent neural activity during real unilateral movement is associated with bilateral activation. We obtained the preliminary results from 11 healthy right-handed participants, 7 females for males from 19 to 28 years old, and surface electrodes. Electromyography was recorded from the abductor pulse brevis, abductor digita minimi, and first dorsal anterosius for both hands, and extensor carpinalis and flexor carpinalis were used as control muscles. Subject performs 3 motor tests: thumb abduction, index finger extension, and little finger abduction. Motor tests were performed in pseudo-random order by right hand, and mirror activity was probed in the left hand. Each motor test consisted of 3 sessions with 30 repetitions of one movement type, 3 seconds isometric contraction followed by 3 seconds rest. Here in the picture, you can see this design that we record EMG from homologous muscles from both hands, and while performing the motor test, focused on the activation of one muscle, we observe in homologous muscles. It's how we hypothesize, but in real life, it can be observed in other muscles too. So, with regard to results, parameters that we measured were persistence, volume, and amplitude. Persistence and where it just appears. Volume is peak-to-peak amplitude. Duration is how long it's appear, and latency is the time between the movement onset, and when mirror activity started. We observed that mirror activity in the left-hand muscles was in 9 out of 12 participants. Mirror activity has preserved in less than 50% of the trials, and PMA tends to increase with repetitions in the experiment course. Average peak-to-peak amplitude of mirror activity was around 150 microvolts, and latency and duration highly varied. It can be during whole movement, or appear only in the end or in the start, or just with duration for several milliseconds. As you can see here, it's just spikes. Thus, our preliminary results show that mirror activity in hand muscles exists in majority of the enrolled subjects. It's highly variable, tends to increase with repetitions, and seems to be muscle-specific. One of the obstacles we faced was that in some subjects in the main hand, they isolated isometric contraction of one muscle without other instigated muscles. It wasn't possible. And to overcome this difficulty, we are going to use pre-training using visual biological feedback for participants. So thank you, our team, for hard working, and our grant who covers federal Russian grant who covers participants. And thank you for your attention. I'm ready to answer your question.
Thank you very much, Ksenia, for this excellent talk. It's really interesting and intriguing. I encourage you to ask questions in the questions and answer box. One of the findings that I found that I think is very interesting is the fact that the PMA increasing with repetitions. So instead of desensitization, it's becoming more sensitive. Do you have any explanation for this?
Yeah, PMA appears in the contral hemisphere, contralateral to the active one, as a phenomenon for coactivation. So when our one hemisphere motor area is activated, there is a phenomenon of leaking of this activation to another. And this is volume of this activation is regulated by interhemispheric inhibition. And as we know, this inhibition is decreasing during while participant with repetitions when a person has less energy. So it's just a fatigue effect. If we continue to repeat or strong contraction, yeah, just fatigue and decreasing of interhemispheric inhibition. And how fast it's appeared and how many repetitions needed to be have a influential impact. It depends on the personal excitability.
Okay, thank you very much again. And we're moving on to the next speaker, Elisa Welly. Elisa is a 5th-year PhD candidate in the cortical neural prosthetics lab at the University of Michigan, and she will talk about Mechanical modifications to carbon fiber electrodes increase recording yield in brain and enable interfacing with peripheral structures.
Yes, hi. Can everyone hear me? Yes, okay, awesome. So hello, my name is Alyssa Welly. I'm a PhD candidate from the University of Michigan in Professor Cindy Chestik's lab. And I'm here today to talk to you about our carbon fiber recording electrodes and the modification of these electrodes for improved brain and nerve interfaces. So this work is based in the field of neuroprosthetics, which translates signals collected from the brain, from nerves and muscles into mechanical movements of external robotic devices, or into the stimulation of existing musculature that may no longer be receiving neural input. And brain-machine interfaces is a well-known technology that incorporates neuroprosthetics. The signals from the brain are recorded, decoded, and then translated into the movement of a computer cursor or robotic hand. Patients like Kathy, seen here, who is left quadriplegic from a brainstem stroke, are able to regain some independence, like being able to feed herself a cup of coffee. So this technology has a potential for a very far-reaching impact, as there are currently 6 million people living with paralysis in the US today. And the crux of this technology, and also its limiting factor, is the electrodes used to record from the brain. So the only FDA-approved cortical electrode is the Utah array, seen here. It's a 96-channel device. It's been around for decades and successfully lasts in patients for years. However, it's fairly well known that conventional silicon electrodes in the brain cause scarring. And our lab was given a rare opportunity to do histology on a single non-human primate implanted with a Utah array for roughly 2 years. And while this data is very preliminary, we can obviously see scarring caused by the Utah array. This is just one slice along the shank, not necessarily at the tip of the shank where the electrodes are. What we're seeing here is endemic throughout. And if you look at the electrode hole itself, one electrode hole, there's an obvious difference between the number of neurons, which are shown here as yellow dots, around the electrode hole as compared to the control tissue. So the data is still being processed. This is very preliminary, but our main takeaway here is that there are just many fewer neurons that we might have otherwise had access to record from. And so the question is, how do we go about maintaining a healthy neuronal population around the electrodes? People have come up with many alternative styles and types of electrodes to solve the scarring problem that we see with the Utah array. Our solution is small electrodes. In our lab, we work with carbon fiber electrodes specifically, each with an individual diameter of 8 microns, once coated with parylene insulation, with a 1 kilohertz impedance of less than 100 kiloohms when coated with PDOT PTS. And so carbon's a really unique material. It's electrically conductive, it's chemically stable, it has high tensile strength, and also very good fatigue resistance. And I'll talk some more about that later. But one of its best properties is its subcellularly sized diameter. And so, you think that maybe these are too small to insert. They're only 8 microns. But because of its inherent stiffness, carbon fiber is able to insert with very minimal assistance from a dissolvable PEG coating. And so we can insert these tiny fibers into the brain as an array structure. And when we reconstruct a 3D image of the tissue around the fibers after they've been implanted for months, this is in rats, we reconstruct it by slice. This video is showing 2 carbon fiber electrodes traced, and you can see them here in gray from 2 angles, and the neurons are in green. And there's a really dense neuronal population right near the electrodes. That you can see neurons, cozied right up near the electrode tips, which means that these electrodes are getting many more neurons to record from for future brain-machine interfaces if we were to use these fibers. So despite this very encouraging histology, there's still work to do. And we noticed that when we implanted these chronically, when we implanted functional electrodes, even though their impedance remained really good, we are actually only recording from roughly 30 to 50% of our electrodes, our functional electrodes, in the brain. So we implanted them and only 30 to 50% of the functional electrodes recorded any neural units. And so, this was confusing because despite the good histology and the low impedance, we're only getting about 1/3 of our electrodes that are actually recording spikes, which is obviously the point of what our electrodes are for. So, in order to use these in future brain-machine interfaces, we want to increase this recording yield to hopefully 100%. And we took a look at our fabrication methods. So there's more about this in our papers, but briefly here, you take a bare carbon fiber, we coat it with parylene C, as I mentioned before. We then cut the tip using microsurgical scissors, and then we electrochemically deposit PTS on the exposed carbon. And when we took a look at these traditional scissor cut electrodes under SEM, however, the results were not great. We saw that the carbon was really inconsistently exposed from parylene. The sites were rough. It was overall highly variable. And, we felt like this was a first and necessary step to improve upon. So we developed a method using blasts from a green light laser to expose the tip of the carbon fiber and peel back some of the parylene to create a more consistent electrode site. You can see some SEMs here of the device or of the electrode with a more consistent and variable and less variable exposed carbon. And so the laser exposes roughly 10 microns of carbon from the tip to create a surface area of like 250 microns squared. And then the impedance drops down to 20 kilo-ohms when coated with PDOT. And so, we've created an electrode that does have a greater surface area than previously seen before. But these dimensions are still on par with an ideal point source electrode. And in fact, the size of a neuron in layer 5 of a rat motor cortex is roughly 15 microns. So we're still, well below the size of a neuron. And, we've created a subneuronally sized electrode with very low impedance. So to test and see if these laser cut electrodes record from neurons and hopefully recorded from more neurons, we chronically implanted 45 electrodes in 4 rats over several months. After 7 days, 84% of the electrodes recorded units. And after 2 months, we were 71% still recorded units, both of which are much higher than the 30% we were seeing from the scissor cut electrodes. So we saw, nearly a doubling in recording yield. But this is not just about quantity, also about quality. And we were, happy to see that the units recorded by the laser cut electrodes were recording amplitudes up to 600 microvolts peak to peak, which, is quite high. And we got, saw that nearly 10% of our electrodes are recording an amplitude that high. And so, with these promising brain results, the natural next step is to translate them into the peripheral nervous system. So right now, again, similar to the brain, the conventional electrodes used in the nerves is the Utah modification of them, the Utah slanted array. And similar to the brain, there's a lot of scarring and inflammation that results from this electrode when chronically implanted in nerves or peripheral structures, and perhaps even more to a severe degree, given this fragile nature of peripheral structures. So, and then also the recording electrodes are so big that recording from like individual axons, where axons are in the single digits of microns in size, essentially it's out of reach to record from individual axons. So, the natural next step is to apply our carbon fiber technology to the peripheral nervous system. So in that process, the first challenge was to be able to actually insert these 8-micron fibers into a nerve. We took inspiration from methods used by the Gardner lab and sharpened the fibers. So the trick here is all about visualization. And I'm going to play this video again so you can see it twice. But we submerged the array underwater in a water bath. The water bath acts as a flame retardant, and we're sharpening them with a small butane flame. And what you're seeing here is actually we're looking at the reflection of the array underneath the water surface so that we can align the tips directly to the water surface so that they can be sharpened by a small butane flame while the rest of the array stays intact. And the ripples of the water that you saw there briefly were the movement of the butane flame. And then afterwards, this is the alignment step. And so that's where the water is moving in response to the flame. And afterwards, you can see a slight tapering in the carbon fiber tips. And so through this process, we get sharpened fibers. There's a really smooth transition between the carbon and the insulation. And although the surface area is much larger than before we were looking at like 10 microns of exposed carbon in length, now we're looking at 140. You'll see that we still are able to record neural activity in peripheral structures. So we sharpened 200 micron length fibers, very short fibers, on our existing brain array for acute insertion and recording from the rat cervical vagus nerve to test this initial sharpened technology. And what we're looking at here is an array of 16 sharpened carbon fibers inserting easily into a rat cervical vagus nerve. The nerve is elevated from its cavity. And then for reference, a rat cervical vagus nerve is anywhere between 200 to 500 microns in diameter. So given that these lengths fibers are 200 microns in length, we can assume they're landing roughly in the middle of the nerve. And as I mentioned, the surface area is much larger than what we used in the brain. However, the sharpened carbon fibers recorded multiple neural clusters from the vagus nerve. And we also tried these arrays in feline dorsal root ganglion and found that they inserted easily despite the thicker covering of the sheath, and recorded neural clusters in response to brushing. So with all that, one of the next things was that the main limitation of the arrays that I showed you before is the fact that the fibers break really easily at the end of the printed circuit board. So to counter that, we embedded the device partially the fibers in silicone. And the silicone greatly increased the robustness of the carbon fibers. Here you can see glass capillary running over the free end of the fibers without breaking. We went up to 50,000 cycles. There was no breakage at that we stopped then because there was no breakage. But this shows the good fatigue resistance qualities of the carbon fibers I mentioned before. And this is something that is very useful when talking about chronically implanting these devices in peripheral structures where there's lots of movement within the body. Combining these things, we've created a new array appropriate for chronic recording from peripheral structures. It has fibers partially embedded in silicone, and the top of the interface is only 1 millimeter by 2 millimeter in size. So, it doesn't look pretty, but it works. And these sizes make it really suitable for recording even from very small nerves in rats. And so finally, we tested the recording ability of this new array that combines silicone and then the sharpened fibers. With our acute device, we were able to implant to a depth of 1.2 millimeters in the rat motor cortex without any assistance from the dissolvable PEG. We recorded neural units. And then we also recently just recorded our first acute spontaneous spikes from the DRG using this new technology. And then our next step is to work towards the chronic implant of these devices in both animal models and assess recording and histological viability. So with that, thank you to funding and then to the many members of my lab and Professor Cindy and Tim Bruns' lab. And I will take any questions. Thank you.
Thank you very much, Elisa, for this great talk. I really love the pictures from your 2020 paper. It's really cool. While we're waiting for questions, I should also mention that YouTube viewers could also post questions in the chat window to the right, and we will paste them here. So, okay, we have the first question. What was the longest implantation time in the rat brain while the electrodes were still recording?
Yeah, that's a great question. Yeah, so the longest implantation time using, so this is without the sharpening technique, this is using our traditional one, is up to several months, so up to 3 months. And then previously up to, 3 months is the longest that we've done. And at the end of that, we were still recording units and we sacrificed due to histological metrics and timelines. Yeah.
I had a question, a couple of questions actually.
Okay.
So how much of the area does it record from? So how local is it, or how broad is the signal that it captures?
Yeah, that's a great point. So I mean, typically the recording distance or the recording area decreases kind of 1/r, so 1/r distance away from the recording. So we typically say that, we want to record from neurons within 50 microns of where the recording site is. And so obviously having a much larger surface area means that we are averaging across a much larger area, right? So if we have a huge, 140 micron surface area, like you saw in the sharpened fibers, we're recording from, a huge volume around that exposed electrode. So you know, the ideal point source electrode would be a single dot, and we're able to record exactly from that space. So we definitely are working towards being able to modulate how much exposed surface area there is for the sharpened carbon fiber so that we can get a, you know, smaller area. However, we also were encouraged to see that we were able to record, you know, neural clusters, maybe not neural units, but neural clusters from the nerve, which means that, you know, we are getting, you know, accurate and high content information, even though we are most likely, you know, recording from a much larger volume.
Oh, thanks. Also, like, can you have multiple contacts on the same electrode? Like you have those silicon U probes, right? Is that possible?
Yeah, so yeah, that's a good question. So each carbon fiber acts as a single wire. So it really allows for just like one contact at the end of it. But there are ways there, and people in the past have worked on ways to like bundle them together to create a structure that has multiple fibers, which can have, you know, different lengths and things like that. Yeah.
Thanks.
Thank you.
Okay, we have two more questions. The first is again from Ksenia Kirilova, who asked the previous question.
Okay.
Is this flexibility of the electrodes that you showed going to be enough to account for brain movement inside the skull during chronic implantation? And how many channels maximum were you able to implant in one animal?
Great questions. Yeah. So the number of channels is up to 16 right now. So that's in the brain. We've done up to 16 and at one time. And then in terms of the flexibility, yeah, I think that when we implant them in the brain, we do cement them to the skull. So the micro movement, or like the micro motion that we'd see, is mostly, you know, the most of that is absorbed in the actual array itself and not the fibers. The fibers are implanted into the brain, and so then the actual like flexible polyimide device that the fibers are attached to are kind of what captures the movement of that like brain micromotion. However, the flexibility that I showed with the silicone embedding, that's going to be more useful for the peripheral nerve interface, where we have the only thing inside the nerves is the carbon fiber itself. And so as you know, there's maybe some movement between muscles or in, you know, kind of the twisting or shifting of the nerve itself, the fibers will be able to bend and accommodate that without breaking. And so that was kind of the main discovery of having the silicone, is that we can have the actual electrodes themselves are able to bend and move and still be robust and not break in response to that movement.
Okay, we have one more question from Don Taylor. Are you working with manufacturers to make these available for purchase?
Yeah, so there's a distribution grant that we have for the brain electrodes, and that's through the NeuroNext Mint Hub. And so if you want to contact, you can contact Paras Patel if you're interested in learning more or receiving those devices for the brain electrode specifically. Yeah.
Okay, I think we reached the end. Thank you all very much, both speakers and
thank you.
attendees. I really enjoyed your talks, and thanks a lot. See you later.
Yeah, bye.
Bye-bye.
The prevalence of ASD has been increasing at quite an alarming rate over the past few decades, actually. And as you can kind of see here, in 2014, this rate was 1 in 59, so very, very high. Another interesting fact about ASD is that males are at a much higher risk of being diagnosed with ASD, being roughly 4 times more likely than females. So the current prevailing opinion on ASD is that ASD is not solely a result of genetic factors, nor solely a result of environmental factors, but actually a combination of factors from both types. And our lab specifically looks at abnormal lipid signaling, because lipid signaling can actually be affected by both of these environmental and these genetic factors. So a little bit more on lipids here. They are a major component of the brain, making up the majority of the brain by weight, and they serve as a cache of signaling molecules that are released when they're required. The lipid we focus on in our lab is the molecule prostaglandin E2, or PG2 for short, and it's one of the more prominent signaling lipids within the brain. So we know that PG2 is involved in neuronal development, but also in synaptic plasticity. So on the side here, I've included the PG2 synthesis pathway. Arachidonic acid is converted into PG2 through the action of this cyclooxygenase 2, or COX-2 enzyme for short. What's interesting about this pathway in particular is that an assortment of different environmental factors can impact it. Things like air pollution, NSAIDs like acetylsalicylic acid that everybody's very familiar with, as well as other consumer products and over-the-counter drugs can impact different parts of this pathway, either increasing or decreasing overall levels of PG2 in the brain. Our lab in particular is interested in the downstream effects that these increases and these decreases in PG2 can have. To study this, we use an in vitro cell model that's exposed to PG2, as well as 2 in vivo mouse models, one in which we expose pregnant dames to PG2 through injection and study their offspring, which serves as our high PG2 mouse model, as well as another model in which we reduce the levels of COX-2, our COX-2 knock-in mouse model, which serves as our low model. So downstream, we've seen that changes, these ups and downs in PG2 can cause changes in gene expression that cause these functional changes in things like migration, proliferation, differentiation, and synaptic plasticity. More interestingly, I think, we've also seen behavioral manifestations of these ups and downs in affected mice, which have shown social deficits as well as repetitive and anxious behaviors, all things that in humans have been used to diagnose ASD. For the purposes of this study, though, I'm just going to be talking about this COX-2 knock-in mouse model. So the primary objective for this study was to investigate differences in dendritic and dendritic spine development in wild-type and COX-2 knock-in male and female mice in the cerebellums at post-natal day 25, with the hypothesis that a COX-2 knock-in would disrupt the normal development of these dendrites in their spines, resulting in abnormal morphology, and of course further that these effects would be modulated by the sex of the mouse. To examine dendritic morphology, we used a technique called Golgi-Cox staining. And so with this technique, we can specifically actually stain neurons and their dendrites, allowing us to see them in very detailed ways. For this presentation, I'll be talking about findings in dendritic arborization, which we measured using shoal analysis, axonal loop formation, which I'll talk a little bit more about later, dendritic spine density, which is just the number of spines per length of dendrite, and lastly, spine shape and morphology, which we classified based on length and width measurements, and then looked at the odds of each spine being a mature shape, so to simplify, just mushrooms, or immature, one of the remainder. So the first trait I'm going to discuss is actually dendritic arborization. So to quantify this, we use a technique called shoal analysis. For those of you who are unfamiliar with this technique, a program will create or draw in circles, concentric circles that are 10 micrometers apart from the center of the soma outwards. And the number of intersections that the extensions of a neuron make with these circles give us an idea of how branchy a neuron is at that distance from the soma. Looking at the graph here, we have the distance from the soma along the x-axis, as well as the number of intersections these neurites make with the circles along the y-axis. Then we have males in blue, females in red. And to differentiate our wild type and our COX animals, I've used a darker color for the wild types and a lighter shade for the COX-2 knock-ins. We firstly noticed that there was no actual sex difference in terms of dendritic arborization. However, we saw a dramatic increase in the number of branches of these COX-2 animals at this 20 to 40 micrometer range. We actually saw that this was reversed once we got to the 60 micrometer length, with COX-2 animals having fewer branches. And I think it's important to note that while there's no significance beyond this point, we actually did continue to see, in terms of the numbers, that COX-2 animals had fewer branches. So changes like this in dendritic arborization can actually have an impact on overall brain shape, as well as in the formation of synaptic connections. And these types of changes are actually usually a good sign that there's some kind of dysfunction in axonal navigation. axonal looping. So when we think about neurons and how they look when we stain our slices, we normally think of neurons looking like this, with extensions that curve and turn yes, but that tend not to turn back on themselves or their dendrites very often. When we were looking through our slides, we noticed that while the majority of them, yes, did tend to look like this, we actually found a decent amount of neurons with dendrites that would loop back on themselves or form these what we call axonal loops. In order to kind of quantify what was going on here, we measured the exterior angle of all of these dendrites. And when we plot all of these measurements with turning angle on the y-axis and then separated out by condition on the x-axis, again with males in blue and females in red, you can see the distribution of these turning angles. What we did then was we classified any dendrite with an exterior angle greater than 270 degrees, so when it turns beyond that 270 degrees, as looping. And when you separate these points out based on that 270 degree distinction, just looking at the top, for example, you can visibly see that there's a difference in the spread of these points. To statistically examine this distinction, we used logistic regression, looking at the odds that a random dendrite picked out of this pool would be classified as looping or non-looping, given its condition and given the sex as well. So matching what we see visually, we can see that sex had no effect on the odds of a dendrite forming a loop, but that when we look at COX-2 animals, that they were highly more likely to form a loop. The degree of this increase in the COX-2 mice we found suggests a dysregulation in kind of axonal pathfinding again, and potentially even more so, cytoskeletal dysregulation, both affecting, again, axonal migration and ultimately the formation of synaptic connection in these mice. When we moved on to look at dendritic spine, the first thing we examined was dendritic spine density, in which we measured the number of spines per micrometer of dendrite on the y-axis here, and then again we have condition on the x-axis split into males in blue and females in red. We saw a surprisingly great difference in spine density between just our wild-type males and females, with wild-type males having a significantly greater spine density than females. Interestingly, in our COX-2 animals, we no longer saw this difference. So given that these changes in dendritic spine density overall are often associated with different neurological and neurodevelopmental disorders, this loss of a sex difference, while it's no longer there, may actually correspond to, we think, larger scale physiological and behavioral changes. Now, looking at density is great, but we know that dendritic spines, they tend to have different shapes that correspond to their function, right? So dendritic spines, they change shape throughout development, with more mature spines typically being mushroom-like and contributing to synaptic connections, whereas less mature spines tend to be more transient in their shapes and tend to have these thin, these stubby, these filopodia-like shapes, and are in general less involved in these connections. So we classify dendritic spines into shapes based on length and width measurements, and then we use, again, a logistic regression to examine if within our conditions, any of these conditions or these sexes would be more or less likely to have mature or immature spines. What we found was that wild-type males were actually more likely to have mature spines, whereas females were more likely to have these immature spines. We saw a complete reversal in the COX-2 animals, with COX-2 males more likely to have immature spines and COX-2 females more likely to have these mature spines. So spine profiles, which is to say the proportion of different spine shapes at different ages to one another, are often again associated with different states of neurodevelopmental disorders as well as neurological disorders, suggesting that these COX-2 mice, given the dramatic switch from the wild-type condition, may also exhibit physiological or behavioral abnormalities again. To conclude, I've included the findings I discussed today into a table with the measures over here, as well as the kind of a summary of the effects we saw. So with regards to dendritic outgrowth, our findings suggest to us that the COX-2 knock-in affects axonal pathfinding and potentially results in some kind of cytoskeletal dysregulation, with the implication, of course, that these mice may be improperly forming connections within their cerebellums. Our spine findings have shown us that there are sex-specific effects of this COX-2 knock-in, resulting in different spine profiles in these mice. So these spine profiles may correspond to improper formation of synaptic connections throughout development. And to reiterate again, in literature, these differences in spine profiles are often correlated with neurodevelopmental disorders and neurological disorders. Given that our study has specifically examined the cerebellum, it seems likely to us that these changes in dendritic outgrowth and these changes in spine profile suggest that these mice, these COX-2 mice, may have deficits in cerebellar-related behaviors, with specific sex differences, of course, in the spines. To wrap up here, I'd like to, of course, thank Dr. Crawford as well as the rest of the Crawford lab for their support, as well as these two facilities here for helping with the slicing, the staining, and the imaging of our slides. And of course, lastly, our funding agencies as well. I think at this point, I'd be more than happy to take any questions anybody might have.
Ashby, thank you so much for your talk. It's very, very interesting and very comprehensive study. So I think we might have a time for one question. So far, I don't see any questions in Q&A box, but I have a question with regard to, so what do you think underlies this sex differences in some differences in morphology, but not the others, specifically the difference in the spines, but not so much in the length of the branches and branching itself?
Yeah, so I think, so from what we've seen so far, it just may be the trafficking of different elements. So PG2 itself is synthesized and can be synthesized in a very sex-dependent way, and its localization, as well as what it acts upon, may be just regulated differently within these axons, either to growth cones or other places along that dendrite as well. So I think that might be part of the reasoning.
Mm-hmm. Mm-hmm. Yeah, that seems plausible. Okay.
As a question, do we still have time? Oh, how to understand the axonal looping? Like what happens there?
Yeah, so that one, so from what we've seen is that typically when you have very dysregulated actin polymerization, or is kind of one reason why you might have these looping occurring. Another one is that there might be the inability or improper ability of these growth cones to receive proper navigation cues. So we're not sure, based on our findings, of course, which one it is. I suspect anyways, that it might have more to do with cytoskeletal dysregulation, given some other findings we have with regards to PG2. But I think, yeah, so I think that's probably what's going on there.
Thanks.
Mm-hmm.
Okay, thank you so much, Ashby. And if there are more questions, you can still ask in Q&A, and Ashby can answer you in the chat. So we will now move on to our next speaker. So our next speaker is Shen Han. She's a postdoctoral researcher in Wishe Hong Lab at UCLA. And in the lab, they're doing a very fascinating work on how brain shapes our social interactions. And she's using microendoscope and calcium imaging to explore how neurons in dorsal medial prefrontal cortex represent sex-specific cues. And she published a paper in Neuron recently. If you want to learn more about this work, you will be able to find the link in the chat. And finally, Shen is on the job market right now, searching for a faculty position. So if you know anyone who's interested, or if you know who is hiring, please contact her. And with that, I'll give the stage to Shen. Please go ahead.
Thanks, Katri, for the introduction. I'm Shen Huang, a postdoc from Wishe Hong's lab at UCLA. I'm glad to share with you our recent exciting work about how sex information of a social partner is encoded or represented in the prefrontal cortex, and how those sex representations shape animals' behavior. Social behavior is essential for animals' health, survival, and reproduction. Many types of social behaviors are evolutionarily conserved, so they exhibit similar features across species. And such behaviors include social affiliation, social dominance, mating and parenting, etc. Many types of social behaviors have a preference for partners of a specific sex. For example, dominance behavior is usually observed between animals of the same sex, while mating behavior is usually observed in animals of opposite sex. So you can see that animals display very different types of behavior, which is dependent on the sex of the other animal. Therefore, being able to recognize sex of other animal is critical for animal to make behavioral decisions. So how is sex information processed in a rodent brain? Those chemosensory cues, such as odorants and pheromones released by other individuals, are mainly processed in main olfactory system or venonasal system. And the two systems converge onto a region called medial amygdala, where sex-specific neural response start to show. And then they further diverge into different hypothalamus centers to trigger sex-specific behaviors, such as aggression, mating, and parenting. However, little is known about how sex is encoded beyond those subcortical regions. Cortical circuits are thought to be more plastic and adjustable than subcortical regions, since they can quickly accommodate outside changes and allows for adaptive behavioral decisions, such as dominance and sexual behavior. In addition, it has been shown by our lab and other groups that medial prefrontal cortex, or MPFC, is involved in sex-dependent behaviors. MPFC is also known for its essential role in social cognition and decision-making. Therefore, we would like to investigate how sex is encoded in this area. The main approach we applied is calcium imaging using miniscope, which is a small version of fluorescence microscope with a super lightweight and has single-cell resolution. The small animals can wear this scope while they are freely behaving, so we can monitor their neural activity during certain type of behaviors. So this shows an example of miniscope recording. We usually can image hundreds of neurons in a single field of view. Through downstream video processing pipeline, we can extract the activity trace of each neuron and align them with behavioral events to analyze how a particular neuron is responsive to a certain behavior. The first question we wanted to ask is whether MPFC neuron encode male and female specific information. So during miniscope imaging, we presented each subject mouse with multiple novel males, novel females, and a toy mouse in a random order. The subject mouse can freely explore each novel intruder for about 20 seconds. And after the whole session was done, we extract the single cell activities and classify cells into categories of female responsive, male responsive, toy responsive, or unresponsive to any stimulus. Here shows some example cells that have a selective response to either male or female investigation. Next, we quantify the number of male or female responsive cells. In female animals, we observed an equal fraction of neurons that were excited by a male or a female social partner. And interestingly, male animals have a larger fraction of neurons that excited by female investigation. This finding is quite intriguing as it suggests two things. Number one, the encoding of sex information in MPFC differed between male and females. Number two, male animals have a stronger representation of female information than the male information. Next, we want to take a look at how sex is represented at the population level, which should give us a more stable and reliable response than individual neurons. We first simplify the data using principal component analysis, or PCA, which is good at transforming a large set of variables into a smaller one that still contains most of the information. The response of hundreds of neurons were mapped onto this three-dimensional space defined by the top three components. Take this animal as an example. it showed a clear separation of population response during male versus female interaction, suggesting that the population activity can distinguish sex information. In addition, we found that male animals showed a higher amplitude of population response during female investigation compared to male investigation, which is consistent with the previous finding that I showed that male animals have a larger fraction of neuron encoding female information. In the following analysis, we focused on male animal to gain deeper insight into this asymmetric encoding of sex information. So why males have a stronger encoding of females, and whether the stronger encoding of female has anything to do with their behaviors. We looked back behavioral experiment videos and wanted to see whether animals have a preference for partners with a specific sex. We measured the time that animals spend with each social partner and calculate the sex preference score, with positive number meaning a preference for interacting with males and the negative number meaning a preference for females. We observed a group level bias toward interacting with females, raising the possibility that the female encoding bias may be linked to this female behavior bias in male animals. Although they show a female preference in average, a substantial fraction of males show a preference for interacting with male animal. So where this individual variability of sex preference come from? And we hypothesize that individual sex preference may be due to its own asymmetric encoding in the MPFC. To test this hypothesis, we first grouped individual animals into female preferring and male preferring groups based on their preference score. We found that the female preferring animals showed a much stronger response during interaction with female social partners than with male partners. In striking contrast, male preferring animals showed a stronger population response during interaction with male animal. This result suggests that the relative activity of male versus female trigger response can predict sex preference behavior. And this will explain the individual variability in sex preference. To test whether there is a causal link between cortical sex representation and sex preference behavior, we would like to manipulate the activity of sex encoding cells and see whether that can influence animals' behavior choice of interacting with male or females. However, it has been difficult to specifically manipulate those sex encoding cells as they typically do not have a simple biomarker, such that an activity-dependent labeling strategy is necessary in this scenario, which can specifically label and manipulate those neurons based on their natural response to a different sex. The particular construct we used is driven by a synthetic promoter called ESER. Here, we adopt the ESER activity-dependent labeling coupled with optogenetic stimulation by injecting two viruses, an activity-dependent Cre recombinase and the Cre-dependent channelrhodopsin. Neurons that were naturally activated by a male or female social stimulus would express channelrhodopsin down the road, so we can use optogenetics to stimulate those native sex ensembles. And this expression process can be switched on by tamoxifen injection. This diagram shows how we carry out the experiments. Each subject mouse was given 1 hour to freely interact with novel male or female in their home cage. After 1 hour social exposure is done, we systematically injected tamoxifen to activate Cre recombinase. A few weeks later, we test the subject mice in a 2-chamber assay where subject mouse can freely choose to interact with either male or female partners. We shine blue light in the middle of the experiments to stimulate the sex encoding ensembles and see whether that makes any changes on sex preference behavior. The control group, okay, got the time. The control group was not given any social stimulus and they were otherwise handled in the same way as the experimental group. We used optogenetics to stimulate neurons that were naturally activated by female cues. Stimulation of female activated cells in the middle of 10 minutes resulted in an acute change in sex preference toward female animals. And in contrast, stimulation of male activated neurons dramatically reversed an overall female preference to male preference. And you can see that after stimulation, they go back to female preference. And this suggests that sex preference behavior is modulated by the activity of native neural representation of male and of female cues. So let's take a summary. I have shown that different sets of neurons in the MPFC respond selectively to male and female cues, and they elicit different population patterns. And the population response amplitude can predict their sex preference. And then when we stimulate those native sex responsive cells using optogenetics, they can directly modulate animal sex preference behavior. And sometimes they can even reverse the original preference to the other sex. This demonstrates a causal role for activity of social representations in shaping social behaviors. And this finding demonstrated for the first time that sex information can be represented in your cortical region. So this will allow for more adaptive behavior decision to accommodate outside and internal changes. Thanks, thanks everyone, and welcome questions.
Okay, thank you very much, Shen. I think it was very fascinating study, especially the part that you were able to actually manipulate the social specific cells and change their sex preference or change their behavior towards the other sex. So that was very interesting. And I think we have time for just a quick question. So if I don't see any questions in Q&A box, but please ask questions. So the question that I have is whether do you have any idea what type of cells those are within the prefrontal cortex that are responsive to the sex-related cues?
Good question. So we did differentiate the chemokines two, like the excitatory cells and inhibitory neurons, but we didn't find a difference between them. So both excitatory and inhibitory cells, they can encode sex information. And there's not a number difference, but it seems that the sex information is more enriched in the inhibitory neurons. As if you normalize them into the same number of neurons, then inhibitory neurons contain more information about sex. But based on other literature or other groups of single cell sequencing, there seems not a difference in molecular composition of the cells. So it's hard to tell what type of subtype of neuron will encode sex. It seems like it's well distributed across different types of cells.
I see.
Great question.
Very interesting. Okay, thank you very much. I think that's all the time that we have for the questions, and you can reach out to Shen on Twitter if you have further questions, or you can ask on Q&A panel. So now we're going to move on to our last speaker of the session, Evan Fullerton. So he's a PhD candidate in Anne Murphy's lab at Georgia State University. And he studies the impact of age, sex, and pain on mu-opioid receptor expression in and binding in periaqueductal gray. That is a critical area for modulation of pain. And so he recently published a paper on this topic. And if you're interested to learn more, you can find the link in the chat. And with that, Evan, please. So Shen, if you could please stop sharing your screen, and then we can move off to Evan.
Yep. Okay. So can everyone see my screen and hear me?
Yes.
Perfect. All right. So thanks for the introduction. The title of my talk is The Impact of Advanced Age, Sex, and Chronic Pain on Opioid Potency and Signaling in the Midbrain Periaqueductal Gray. So in our lab, we study chronic pain, which is a prevalent and debilitating condition. And indeed, 1 in 5 Americans suffer from chronic pain, which is defined as pain lasting greater than 3 months in humans. And one of the variables that we look at in chronic pain is how advanced age affects chronic pain suffering. As you can see from this graph, individuals are more likely to suffer from chronic pain as their age increases. And in addition to advanced age, we also look at biological sex. As you can see from this graph, females are more likely than males to experience chronic pain. And those differences, the sex differences, are even greater as we look at the higher age ranges. And so we study sex and chronic pain and as well as age. And part of the reason this is so important is because the American population is aging. As you can see from the right side of this graph, the demographic of Americans representing those 65 years and older is modeled to represent nearly 25% of the population in the coming decades. And despite the fact that aged individuals experience chronic pain at higher rates, there's a need for advanced age and opioid analgesia studies. And this is because pain in aged individuals is commonly undermanaged. So even though opioids such as morphine are really common treatments to use to manage chronic pain in most individuals, there exists a dearth of knowledge regarding the suitable dosing of opioids for chronic pain management in the elderly. And so our studies use a laboratory model using Sprague Dawley rats to assess the impact of age using 2-month-old adults and 18-month-old aged animals, as well as sex using both males and regularly cycling females. And what sets our research apart is that we use a clinically relevant model of chronic pain in the form of complete friends adjuvant, which is an inactivated mycobacterium that, when injected into the hind paws of our animals, creates localized swelling and inflammation, as well as hypersensitivity to pain. So this injection is used to model the pain that a human would experience after an injury or surgery that would lead to a morphine prescription. And so we give our animals CFA, and our animals are in a state of chronic pain, and we ask first, how do age and sex impact morphine potency? So we assess morphine potency by looking at the sensitivity of an animal to a noxious thermal stimulus, or a Hargrave's test, where we shine a radiant light beam on our animal's paws and measure the amount of time it takes these animals to reflexively withdraw their paw. And then we give our animals injections of subcutaneous morphine. So it's a systemic dose of morphine. And throughout the period of several hours, we subject our animals to a cumulative morphine dosing paradigm, where every 20 minutes, the animals are injected morphine and their behavior is measured following this injection. And so their dose accumulates into 18 milligrams per kilogram of the animal's body weight, and we generate dose response curves on how morphine brings up analgesia. And so you can see the result of this dose response curve here, where the x-axis is morphine concentration and the y-axis is morphine efficacy, basically. And so this rightward shift that we see with our adult females, aged males, and aged females shows that these animals, when compared to our adult males, require higher doses of morphine to reach adequate analgesia. And we generate values from these curves in the form of EC50 values, or effective morphine concentration. Basically, this is the dose of morphine that these animals need to effectively manage their chronic pain. As you can see, it's higher for our females and for our aged animals. And we assessed this using an ANOVA to make sure it was statistically significant. Okay, so this table that you're seeing, these EC50 values, are how much morphine an animal needs systemically to modify chronic pain. And we're looking at these in terms of morphine potency values. So we're using our adult males as our baseline and looking at how we see shifts in morphine potency in our adult females, aged males, and aged females. And so this behavioral information leads us to our question of, by what mechanism do age and sex alter opioid potency? So for some brief review on pain signaling and morphine analgesia, basically peripheral pain is sent centrally along this ascending spinothalamic tract from the spinal cord up until the cortex and thalamus. But incoming pain signals are negatively modulated or inhibited by this innate descending modulatory tract. And critical to this descending modulatory tract is our brain region, the midbrain periaqueductal gray, or PAG. And this region is important because it's the center for downstream modulation of pain, and also because it has a dense expression of mu opiate receptor, or MOR. So our mu opiate receptors are the proteins to which exogenous opioids such as morphine bind to bring about the inhibition of an incoming pain signal. And previous studies have looked at the population of mu opiate receptors within the ventrolateral PAG and found that it's critical for opioid analgesia, or morphine managing chronic pain, where intra-PAG mu opiate receptor agonists bring about analgesia, and intra-PAG mu opiate receptor lesions and antagonists weaken analgesia. So this information about mu opiate receptor in the PAG and our behavioral data led us to form the hypothesis that advanced age and sex impact morphine potency by altering opioid signaling in the midbrain periaqueductal gray. And we assess this hypothesis with two questions. So basically this talk is broken into two parts. The first one is, we ask, how do advanced age and sex impact the ventrolateral PAG mu opiate receptor expression and binding. And so we assess expression using immunohistochemistry and in situ hybridization, and we assess binding using receptor autoradiography. So for our fluorescent immunohistochemistry, we're looking at protein expression by tagging the extracellular surface of our protein. And we use fluorescent antibodies to determine the intensity and density of our mu opiate receptor stain in our PAG tissue. And so you can see an example of a three-dimensional mu opiate receptor section here. So when we assess group differences in mu opiate receptor expression, we found an impact of age that's primarily driven by our aged males, where we see reductions in mu opiate receptor expression in our aged animals, specifically in our aged males, and we see no impact of sex here. But what we wanted to make sure is that this reduction in mu opiate receptor expression is not due to an overall loss of neurons. So as a control, we ran a UN stain and determined that the age-induced reduction in our mu opiate receptor is not due to reduced cell density. And as another control, we looked at an additional brain region, the inferior colliculus, and again found no significant differences driven by age or sex, suggesting that the age-induced reduction that we see in the mu opiate receptor in the PAG is somewhat specific to this region. So after our immunohistochemistry results, we ran autoradiography. And the difference here is that rather than just looking at where the receptor is, where it's expressed, we're using a ligand in the form of DAMGO, which is a synthetic peptide with a high affinity for the mu opiate receptor. And by tagging our ligand with a radioactive signal, we can look at where our mu opiate receptors are binding and whether we see impacts of age or sex on binding in the VLPAG. And so we generate these heat maps in our midbrain sections, and we can assess the strength of our radioactive signal, which is basically showing us where our ligands will be binding to our mu opiate receptors. And so our results from our binding assays are shown here, where we see an impact of age in that our aged males and our aged females show reduced ventrolateral PAG mu opiate receptor binding. So there's a functional difference in the receptors in our aged males and females. And again, we see no impact of sex. It's primarily driven by age. And the third experiment and part of part one is to assess differences in mu opiate receptor mRNA. And these experiments are currently underway. We're using RNA scope for single cell in situ hybridization. And you can see the OPRM1 gene, which expresses the mu opiate receptor, is here fluorescent green. So we're currently assessing age and sex differences in mu opiate receptor intensity and number of puncta. So a brief review on part one. Basically, with our behavioral data, we found reductions in morphine potency in our females and in our aged animals. And using immunohistochemistry, we found reductions in mu opiate receptor expression in our aged males in the PAG. And using autoradiography, we found reductions in mu opiate receptor binding in our aged males and aged females. This brings us to part two, where we ask the question, how do age and sex impact VLPAG mu opiate receptor pharmacodynamics? And so here we're looking at the relationship between the mu opiate receptor and an agonist, as well as agonist-induced G protein activation, since mu opiate receptors are G protein coupled receptors. So we use our radioligand binding assay and our GTP gamma S assay. And so we're first looking at the binding of an agonist or an opioid to our mu opiate receptor. And as you can see in the cartoon on the right, when we look at our agonist binding, we're assessing differences in receptor availability and differences in our receptor's affinity for an agonist or for a ligand. And when we get agonist binding to our mu opiate receptor, this signal, which will eventually be the signal that inhibits incoming pain, is mediated by G protein activation. As you can see, this inactivated G protein on the left and this activated G protein on the right. And to assess differences in G protein activation, we use a GTP gamma S assay. So for our ligand binding assay, we generate these binding curves for our four groups, our adult males, adult females, aged females, and we can assess the shape of these curves to determine our overall binding potential. And I want to thank Dr. John Stryker at University of Arizona, who lent us his protocol to help us fine-tune our assay for this. But basically what you see in the graph here is the result that we see reductions in overall binding potential in our females and in our aged animals. So our adult males have the greatest mu opiate receptor binding potential. And these results mirror our behavior results of morphine potency, where we see reduced binding potential in our adult animals, or sorry, in our adult females and in our aged males. And we see reductions in morphine potency in these same groups, thus suggesting this reduced binding potential in this brain region contributes to our reduced morphine potency. And next, our GTP gamma S assay, we're modeling
2
morphine.
minutes.
How many minutes?
2 minutes.
Perfect. We're modeling morphine bringing about G protein activation by using DAMGO, and we use a radioactive form of a GTP molecule. So we can measure the actual activity of our G protein receptor, or our G protein coupled to our mu opiate receptor. And so just like with our ligand binding assay, we generate these binding curves for our animals, and we use this curve to generate G protein activation efficiency values. And so the activation efficiency represents how likely an opioid is to bring about G protein activation. And so as you can see from our results, we see reduced G protein activation in our aged animals and in our females. And again, our adult males have the greatest activation efficiency. And again, these values look just like our morphine potency values, suggesting that reductions in G protein activation contribute to our reduced morphine potency. And I'm going to skip the part 2 summary and go right to the project summary. So basically, we found reductions in morphine potency in our behavioral assays. We found reductions in mu opiate receptor expression in our aged males, and reductions in mu opiate receptor binding in our aged males and females. And I'll hopefully have my mRNA data within the next couple weeks. And when we assess the changes brought on by morphine potency, looking at our binding potential with our radioligand binding assay, we see reductions in our adult females, aged males, aged females, and same with our GTP results with our G protein activation. So all of these changes that we see as a result of age and sex are potential targets that our future studies and hopefully future studies from other labs can look into to potentially reverse the aging effect and thus improve the potency of opioids, allowing for aged individuals to receive effective pain management, whether they're male or female. And with that, I'd like to thank my lab and my collaborators and my funding as well.
Right, Evan, thank you so much for your talk. It's also very comprehensive and interesting study showing the difference in both morphine potency and binding across gender. Especially for me, it was fascinating to see that there is a very big difference between morphine potency between female and male rats.
Right.
Maybe leading to different pain management across gender.
Yeah, it's interesting. As far as I know, I think the guidelines are basically only based on the weight of an individual and potentially metabolism. And so usually I don't think that there are very many notable differences in how chronic pain management is given to females versus males, which is, yeah, it's another issue that, we've tried to focus more on the aging issue. But basically, personalized medicine is extremely important, and especially for the management of chronic pain.
Okay. Thank you very much. So we're actually a little bit over time. So I would like to thank all of the speakers. I learned a lot about the gender differences across different branches of neuroscience. So thank you very much again, and thanks for the presentations. Okay, is that it?
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you. I mean, really, thank you. Thank you. Bye.
Bye.
Dan, are we waiting for more, or we can start?
I'm good to go.
Sorry. Hi,
Sorry,
everyone.
I was muted. I think we should just get started.
Okay. We have a great lineup of speakers today. We have Lena King, Akul Gupta, and Harpreet Saini. And tomorrow we can start. Sorry? Did I hear anyone? Okay. Let me introduce Lena King. She's a graduate student in the Cognitive Neuroanatomy Lab of Kevin Weiner in University of California, Berkeley. And she'll give a talk on transcriptomic contributions to the cytoarchitectonic parcellations of the human cerebral cortex.
Awesome. Okay, so I'll go ahead and share my screen here and we can get started. All right. And so just checking everybody's seeing my slides here. Okay, sweet. Just introduced, my name is Lena King. I am a second-year PhD student at the Helen Wills Neuroscience Institute at Berkeley. And today I'll be talking about my current research that is looking to investigate the transcriptomic contributions to the cytoarchitectonic parcellation of the human cerebral cortex. So in order to address this question, the lab utilizes two publicly available data sets. The first one being the Allen Human Brain Atlas that has been shared by the Allen Institute. This is a microarray data set that consists of gene expression values for over 20,000 genes with samples taken across 6 different postmortem brains. The second data set is the Jülich Cytoarchitectonic Atlas, which is a whole brain parcellation of 100 different regions throughout the cortex. This parcellation was conducted by Carl Zillis and Katrina Muntz and their colleagues in Germany, in which they use an observer-independent method to algorithmically define these regions based on differences in cytoarchitecture. So as seen in the figure here, cytoarchitecture is defined as size, shape, and distribution of cells across cortical layers. And crucially, these differences in cytoarchitecture have been linked to differences in function as well. And so also important to note, these two data sets are both aligned to the MNI 152 space, which allows us to easily integrate these two data sets into a single pipeline. So we use a hypothesis-free approach to determine the differential gene expression across 88 cortical cytoarchitectonic RRIs, while there are a different number of microarray samples within each RRI. As in our previous work, we use ANOVA with permutation testing to select the top 1% of genes that are differentially expressed across RRIs. This process results in exactly 200 genes that we focus on for the remainder of our analyses. So the first thing we wanted to look at was how these cytoarchitectonic regions cluster based on the expression profiles of these 200 differentially expressed genes. So using hierarchical clustering, 2 distinct clusters are produced at the highest level as indicated in the stenogram, cluster 2 in orange, cluster 1 in green. And to further examine the transcriptomic contribution to the clustering of these 88 regions, we projected these clusters onto a 2-dimensional space using multi-dimensional scaling. So as illustrated here, same clusters, cluster 1 still in green, cluster 2 still in orange. Interestingly, each cluster contains regions from the same as well as different functional networks. So for example, visual areas are illustrated together, indicated that they are transcriptomically similar, with a distinction between early and late visual area regions, where in this case, we are looking at within network clustering. On the other hand, high-level visual areas in the posterior and anterior fusiform gyrus, shown by FG here, are transcriptomically distinct as they are located in different clusters. So for instance, FG1 and FG2 in the posterior fusiform gyrus are located in cluster 1, while FG3 and FG4 in the anterior fusiform gyrus are located in cluster 2. So we're looking at a cross-network clustering here. And then additionally, let's see, FG4 clusters more closely with STS, which is the superior temporal gyrus, whereas FG3 clusters more tightly with regions in the orbital frontal cortex. And then FG1 and FG2 regions cluster with parietal and cingulate regions. So to conclude, we consider these present results in the context of previous results, examining the relationship between transcriptomics and either functional maps within areas or the positioning of areas within a cortical network. So for example, previous work by Gomez and colleagues show that a sparse set of 200 genes generated orthogonal transcriptomic gradients that contributed to the positioning of areas within the visual processing hierarchy. Additionally, work by Zen and colleagues shows that orthogonal gradients also contribute to the layout of orthogonal functional maps within visual areas. And what's interesting here is that different sets of genes contribute to the layout of functional maps within areas and the positioning of areas in the visual processing hierarchy. while 60% of genes identified in the research that I just presented here are shared with those genes contributing to the layout of the human visual processing hierarchy. So taken together, we propose that the combination of these findings across studies suggest a nested spatial relationship between transcriptomic, anatomical, functional, and functional features of the human brain at these different scales. So at the micro level, the meso level, and the macro level. And so finally, we propose that despite the fact that different genes contribute to these different scales, cellular density is likely a parsimonious explanation linking transcriptomics to the cortical layout of functional maps within single areas, the arealization of processing hierarchies largely constrained to a single lobe, and then the arealization of the cerebral cortex across all 4 lobes as well. And so with that, I just want to say thank you to my PI, Kevin Weiner, as well as Jessine Songley for the help and all their contributions to this project and the work, and as well as their current work, and also to NeuroMatch for hosting me.
Thank you. Thank you very much, Lena. While we're waiting for questions to come in, let me ask you a question. It might be a lay question because it's not exactly my field, to say the least. Can you, or how can you justify or verify or test functionally this percolation afterwards, like with imaging or other techniques?
So I guess there's 2 ways I can answer this question. So one way is looking at a single gene level and seeing, what functions that gene is associated. So with these 200 genes that we pull out, we can use the gene ontology database to get, their specific function or where they're found out in the brain or how they work, how they're made. Or another thing that's been done is with this work that was by Gomez, which is looking at the relation of genes to both the anatomical and functional hierarchy. The visual cortex has a very well-studied functional hierarchy. And so if we have these different genes that has this very clear gradient, it's a very small figure here that I can actually pull up. So this is kind of enlarging that figure a little bit. But this expression level of these genes vary in a very nice linear ascending and descending fashion. And this correlates really strongly to the hierarchy of, how these anatomical regions are ordered together. So we have HOC1, which is early visual regions, and then we start working our way up to much higher visual processing regions. So hopefully that answers your question.
Okay. Yeah, what I meant was, I guess, more in the systems level.
At the systems level, okay.
Yeah.
I see. So I am also not in the field of systems research, so that is something very much outside my expertise. I'm sure there are definitely a lot of things you can do to test. So taking one of these main genes, for example, in the 200 genes, and then just kind of testing that at the systems level to see exactly what it's doing in the circuit. I think that's definitely a good approach, a good way to combine different disciplines of neuroscience, but.
Yeah, I mean, I guess there is some kind of discrepancy between the 2 levels, right? The function, the system levels is not enough fine or have a high resolution in order to test this more high resolution percolation. So there might be a problem of 2 different levels.
Right. Yeah, it is using these genes very kind of this micro feature in anatomy and correlating it to a very macro level scale. So yeah, I would say it is important to have that intermediate systems level between the 2. So.
Okay, great. Let me ask you, what stage of are you in the publication? Have you sent it? What stage
no.
is this work
This work that I am currently working on is not been sent for publication yet. But currently, the manuscript is we're kind of in the processing or pre-steps, early stages of the manuscript preparation. So still kind of finalizing figures a little bit and finalizing our organization and exactly how we want to present this. But we are in the process of preparing it. So.
You have beautiful figures.
Oh, thank you.
I think this is ready.
This is
yeah. All right. Do we have any other questions from the attendees or from the panelists? Let's wait 1 minute more and then we'll move on.
So there is one question from Jacob Miller. I'm curious if you think that cell density in any specific cortical layers may be more important in terms of linking genetics to functional organization. So we have actually taken a look at this analysis in regards to the von Economo data, which does have layer specific values for things like cellular density. And so I actually have looked at like the layer specific values in relation to this, I guess, like a macro level organization. And it's kind of like the overall value across the different cortical layers that represents more closely this change in cellular density across regions. So it's not really layer specific. When I was looking at individual layers, I didn't like see any strong correlations or anything like that as I did with the overall layer. So yeah, I don't think it is entirely layer specific, but.
Thank you, Jacob, for the question. And I think we can move on unless there are other questions. Let's wait 1 more minute.
I can also stop sharing my screen.
Yes, okay.
There we go.
Okay, let us move on to the next speaker. Next we have Akul Gupta, who graduated from University of California, Santa Cruz, and was working in this project that he's going to talk about in Allen Institute. He's going to talk about recurrent neural network reveal distinct signal flow in mouse visual cortex following expectation violations.
Hi, thanks for the introduction. Yeah, my name's Akul. I was an intern at Allen Institute working with Farzaneh, and we were interested in seeing how, studying cortical column interactions in the mouse visual cortex using RNNs in response to novel images. So let me first start off with motivation. So we wanted to get a better understanding of the predictive coding framework. So quickly, the predictive processing framework speculates that the brain makes predictions about external inputs and updates these predictions when they don't match the inputs. However, it's not clear right now how different cortical areas and at different cortical depths interact with each other to represent and update these predictions. So here we wanted to leverage RNNs to get insight into the circuitry that allows predictive coding. So at the Allen Institute, we have mesoscope data. So what that is simultaneous imaging of 8 cortical planes. So we have 4 depths in 2 visual areas, V1 and LM. And so we have 3 mouse lines tagged for excitatory cells, and we have 2 inhibitory populations, SST and VIP. So what the mouse is shown is image every 500 milliseconds. So it's shown image, 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 and then randomly, there's sometimes an omission of that image when the mouse is expecting the image. So we call that an omission. And the last thing you need to know is we have 2 image sets. First is called the familiar image set. So what this is image set. These are images that the mice has seen before it's trained on these. It recognizes these images. And then on a new day, we'll switch to a different image set called the novel image set. These are images that it's never seen before, so they're novel. So the methodology for training the RNN. So the number of units in the RNN is the number of recorded neurons. So if I have 500 recorded neurons, I'll have 500 units in the RNN, and each unit in the RNN is fit to the activity of the recorded neuron. So in total, so this, what I'm showing you here is an omission aligned trace. So this is what I call one trial, and then I'll have around 200 of these trials. And so so over here on the bottom, this is the input to the RNN. This is the input trace. So this is the stimulus information, whether the image is present or not present. And in blue, this is the population average of the recorded neurons. And in orange is the average of the reconstruction of the RNN. So this is the same thing except at the individual neuron level. So these are individual neuron traces, and you can see the RNN is doing a pretty good job of fitting the neural activity. So now I'm going to talk about the RNN weights, which is the results. And RNN weight magnitudes are informative. They tell us about the connectivity strength between different neurons. So the first thing that I'm going to focus on is what I'm showing you here are the input weights to the RNN. So the RNN has two sets of weights, inputs and recurrent weights. So the input weights are the weights from the stimulus information to neurons. So the first thing I'm going to focus on is excitatory weights. So we have 8 planes here. Like I said, 4 planes in LM, 4 planes in V1. And what I'm showing you here is the percent change from familiar to novel. So we're looking at what's changing from the familiar session going on to the novel session. And we can see here the thing that strikes out is in deep LM, we see a strengthening of weights for the excitatory cell line. Moving on to SST, the inhibitory cell, we see across the board decreasing in weight strengths, specifically in deep planes of LM and deep planes of V1. So also this is coming back to biology, and we see that for the novel session, SST has smaller image responses. So this correlates well with what the RNN is finding. And also looking at VIP, we see the opposite effect where we are getting an increase in strengths. Also, we see them particularly in deep LM and V1. So the same thing, the traces in biology, we see that VIP neurons are responsive to images in the novel session, but not responsive in the familiar session. So in the RNN, we see these change of weights reflecting what we've seen in biology. Okay, so those were the input weights. So now I'm going to be talking about recurrent weights. These are the weights going from neuron to neuron, so how different neurons are connecting with each other. And so this column is feedback weights, so from LM to V1, and then this is feed forward, V1 to LM. So first off, in excitatory neurons, we're seeing in the novel session, we're seeing increase in weights from deep LM to V1 and V1 to deep LM. So mutual connections with deep LM are increasing. For SST, the inhibitory, we're seeing across the board decrease in weights with no particular preference on any plane. However, interestingly, we see in VIP, we see an increase in weight from LM to superficial layers in V1. And so this is interesting because in literature, top-down predictory inputs have been studied to come from, to project onto superficial layers. So the takeaways here are that RNNs can reveal functional connectivity changes across cortical areas to novel images. And this is something that we can't do with correlation analysis. So this is really great. Also, I've shown you how different cell types have distinct changes in connectivity, excitatory, increase in deep layers. With VIP, the input weights are increasing across most layers, and recurrent weights, feedback from LM to superficial V1 is increasing. And with SST, we see mostly increase in deep layers for input weights and recurrent weights with no preference. So overall, response to novel images, we see increased excitatory feedback in LM and increased feedback to VIP neurons in superficial, which is important because VIP neurons have been studied and they're thought to carry the prediction signal or prediction error signal. So RNNs give us a further way to study this. So I'd just like to thank Farzaneh, my mentor. She's great help. We've had so many conversations and it's been just really fun working with her. I'd like to thank the Allen Institute for Brain Sciences and the Neuromatch conference organizers. This is a cool conference.
I agree. Thank you very much, Akul. It was super interesting. We have one question from Cole Horwitz. It seems as though the largest mistake that the RNN makes when predicting mean population activity is around the same time as the omitted image. Do you have any intuition about why discrepancy occurs?
So the largest mistake, so over here. Yeah, I guess one thing is that's interesting actually. This is actually just one trial, so it's actually not the case for all the other trials. So it's actually not the case. I guess I could have put multiple figures here.
Akul, do you have another figure with more than one trial?
I don't right at this moment.
Okay.
Let me see if I should.
We still have time, so no pressure. Take your time and give us figures if you have. You don't. Are you looking for other figures, Akul, or?
Oh, yeah. I can look for this, but I won't.
It's okay. Let me ask another question, and then if we have time, we'll do that. Okay?
Yeah, sounds good.
Okay. So Robert Woodry asks, are omission elicited prediction error signals stronger than novel stimulus prediction error signals for your model? Or do they elicit the same strength?
Sorry, can you repeat that? The audio's actually, I think I'll
Sure.
just read the question. I don't know where they are though.
Let me read it. Are omission elicited prediction error signals stronger than novel stimulus? Wait, it disappeared. Stronger than novel stimulus prediction error signals for your model? Or do they elicit the same strengths?
Okay, so I think so our omission elicted signal stronger than novel. Okay, so I see the question. So basically, the cell line that has a distinct response to the omission is the VIP cell line, which is thought to carry the prediction error signal. And so for novel stimulus, the VIP cell line, it doesn't carry that signal. So the difference is when it's the familiar image set, the prediction signal is there. However, when it's the novel image set in the VIP cell line, the prediction error is not there. So once it starts learning the novel image set, then we start seeing the prediction signal. And let me show you that. Yeah, I can't seem to find the share screen button anymore. It's funny.
Sometimes it's hidden. It's like you have to hover the mouse above the lower part of the screen.
Yeah, it's like playing hide and seek with me right now. Where?
Try to bring your mouse to the lower end of the screen. There is an answer for the previous question from Farzana Nafaji. So I'll read it as an answer to the previous question. To answer your question, Akul is right, but more directly, his RNN was trained on a trait that included both images and omissions. He did not test the RNN on omissions only. He plans to do that though. So we don't know if in his RNN the response is more to omissions or to novel images. Do you want to add on this, Akul?
Yeah, I think that perfectly captures it. This is trained on both images and then the omission, and then more images. So I haven't tested it out where I've only trained the omission trace. So it may be that partially that to minimize the loss, that the RNN might be preferring to just respond to images and have that and take the loss on the omission part of the data trace.
Okay, and Dr. Nafaji just continued to answer. Also, regarding the previous question, can you please repeat it? I might be able to help. I couldn't hear well. Unfortunately, we cannot bring the attendees alive, so I'll have to repeat the question. Are omission elicited prediction error signals stronger than novel stimulus prediction error signals for your model, or do they elicit the same strength? Dr. Nafaji, please let me know if you heard this.
In the meantime, could I ask a question to the speaker?
Yeah.
So I was wondering, is there an effect of sort of N back? So how long ago there was the last previous emission was, and task duration, like is there a decreasing surprise effect after a number of emissions?
Yeah, so the first part of your question, where how long ago the previous emission is, that's actually something that I haven't studied or looked into, so I don't have a clear answer for you. But for a surprise effect, yeah, after for the novel image set, once the mice continues to see these images, there is then it starts getting the predictory signal. But in the inverse case, the opposite case where it keeps getting this like omission. I don't believe there is any surprise decrease in response to the omission. So this omission signal still continues to be strong.
Okay. Do we have more questions? We still have 2 minutes before the next speaker. So we have another answer from Nafaji. Akul is right. We have very few emissions, but we think if we had more emissions, we would see the prediction error reduced. Okay. Thank you very much, Akul, once more.
It's a pleasure.
Thank you. And we're moving on to our next speaker. Let me introduce Harpreet Saini. Harpreet is a second-year master's student of biology and neuroscience at York University. And she is going to speak on the color of the flesh attenuates its mislocation in the flesh jump illusion.
Okay, thank you for that, Shlomit. Let me just share my screen. Okay, so Bayesian models of perception can be used to explain and understand how color differentially modulates different perceptual and attentional processes in the visual system. In a Bayesian framework, the strength of a color can bias the weight of its priors, resulting in different behavioral outcomes. And previous studies have found an attentional color hierarchy, which has been shown to modulate various cognitive processes such as target selection, visual search, and response inhibition. However, these color effects have been found for later stage decision-making processes. But in our study, we wanted to investigate whether color-dependent modulations also affect lower-level perceptual processes, such as feature integration, within a Bayesian framework. To test this, we use the flash jump paradigm, which is a visual illusion that integrates apparent motion with color. This perceptual misbinding is proposed to be due to a Bayesian framework. So when you've got an apparent motion sequence of moving bars, one of the bars changes color at a single position along its trajectory. And this color change is perceived to occur farther along the trajectory of the bar's motion at a later occurring bar instead of the bar where it actually occurred on. And this is a mislocalization of feature integration between color and motion. Interestingly, no one has manipulated the color of the flash to see its effect on the magnitude of the illusion, which is what we tried here in this study. So we hypothesized that if perception is based on a Bayesian framework, then we would expect that the stronger the attentional strength of a color, the more veridical the perception of flash location. So we would see reduced mislocalizations. And in our paradigm, participants fixated at a cross for 200 milliseconds, and then an apparent motion sequence of 20 gray bars was presented, which either moved leftwards or rightwards. And one of the central 11 bars flashed one of 4 colors: isoluminant red, green, yellow, or blue. And after the sequence, after that color flash, the sequence returns to gray and it continues. And after the sequence is presented, participants were asked to report the location of this color change by clicking with the cursor that appeared on the screen. And on a subset of trials randomly interleaved, we also presented the terminated condition. In this terminated condition, once this flash or this color change appears, the motion sequence is terminated such that the subsequent bars are not presented. And previous studies have shown that in this terminated condition, there's little to, if any, shift in perceived location of the flash, because there are no later occurring bars to mislocalize this flash or this color change to. So this condition, this terminated condition, served as our control. The 2 metrics that we looked at for this study were accuracy and precision. And accuracy was defined as the mean shift between the real location of the flash and the perceived location of the flash. And precision was the trial by trial variability in the perceived location. And we looked at both these metrics for all 4 isoluminant colors. So I'll begin with the results from our accuracy analysis first. Note that positive values correspond to a forward shift or forward mislocalization along the direction of motion of the bar. And as expected, we found little to no shifts in the terminated condition, which was the control, and very large shifts in the continuing condition. And more importantly, when we looked within the continuing condition, we found that the magnitude of the shift varies depending on the color of the flash. Here we saw that red and blue flashes were less shifted than green and yellow. And similarly, for precision, we found that red and blue flashes result in more precise predictions of flash location compared to green and yellow. And that matches the pattern of effects that we found for our shift analysis. So in conclusion, here in our study, we've shown that participants demonstrated improved accuracy and improved precision for localizing red and blue flashes compared to green and yellow. And this is further support that perception and feature integration follows a Bayesian framework, because the colors that we've tested for were all isoluminant. So we shouldn't have seen any differences in the magnitude of the illusion based on color. However, our results here have shown that the color of the flash biases the strength of the priors, resulting in different levels of mislocalizations based on the inherent strength of the color. In our case, red and blue produce stronger priors than green and yellow. And lastly, based on previous work by Stenberg et al., we would expect to find these color-dependent modulations of the flash jump effect in V4, but that would require further neuroimaging or recording studies. Okay, and I'd like to thank all you guys for attending, and thank you to my lab and my lab mates. I'm open to any questions that anyone may have.
Thank you very much, Harpreet. Yes, we're waiting for questions. One question here from David Johns. What do you expect to see if the color change occurred at the start of the sequence?
Okay, so give me 1 second if I can. Okay, so if, I guess the question is if the color change occurred at the beginning of the sequence instead of occurring at one of the central 11 positions, I would expect that we would still see the flash illusion. We would still, the effect will still persist because previous studies on motion-based induced or motion-induced position shifts have shown that this effect still persists even if the flash is occurring at the beginning of the sequence or somewhere in the middle, but it doesn't persist if the flash was the last occurring bar in the terminated sequence. So if this flash occurred at the first bar, given that color and motion are processed in different systems and processed at different rates, the color information would be processed slower, so then it would be mislocalized or misbound to a later occurring bar somewhere down the sequence. So I would probably expect that this effect would still persist. Okay.
Hey, another question here from Nikki, which says, good presentation. You mentioned that previous studies have shown a hierarchy of color differences. Is there a reason why both red and blue are more accurate instead of only one color? Okay.
So I think the effects that the pattern of color effects that we're seeing here could be related to color opponency. The colors that we have tested for are all opponent colors. And so red versus green is an opponent pair, and blue versus yellow is the other opponent pair. We're proposing that red and blue are the stronger colors out of their respective opponent pairs, and that information gets fed into V4 because V4 receives color opponency information. And previous work by Sundberg et al. Have found neural correlates of this flash jump illusion in V4. So we're proposing that this color dependent modulation is probably due to color opponency information that's feeding into V4. I hope that answers their question.
Okay, thank you. Are there other questions?
Oh, I have a question about, you mentioned these colors were equiluminant, right?
Yep.
Do you think it matters whether you move from a white stimulus, which is more luminous, down to one of these? Because I seem to notice that like yellow and green are typically, as we typically see them, are more luminant than red and blue.
Right. We haven't tested. So are you suggesting that the flash is a white color and then the rest of the bars are gray? Is that, did I interpret that?
I don't know what your stimulus, I thought it would be white and then suddenly a color.
So the rest of the bars are gray, and they're also matched in isoluminance to the colors of the flash bars. So everything is isoluminant. The gray bars and the flash bars.
Right. I'm sorry, I misunderstood.
I also have a quick question. So in your subjects, you have 24 subjects. Did you ever see a case where it was switched, or was it a very consistent across all 24 subjects that red and blue were, yeah.
Okay, so you're saying how consistent was this pattern across 24? This particular pattern, I looked individually, and this was consistent for 21 out of 24 participants. So I would probably guess that's pretty consistent. And just participants, the pattern was switched, I think, for one of the colors.
Right. Yeah, I'd be curious to see if there are certain people with like specific types of color blindness or like color blindness or like color perception that this case might be switched. But yeah.
So actually, we test for color deficiency before we do these experiments by using Ishihara plates, so like a quick test for color deficiency. And if they don't perform well on that test, then we exclude those participants from this study.
I see.
Thank you. And David Jones just followed up with another question. Thank you for the answer. I am familiar with the flash lag effect. Is this flash jump illusion similar to the flash lag effect?
Is this flash jump? Okay. So yes, flash lag is probably the more well-researched and well-studied of these types of illusions, but they're both fall under the category of motion-induced position shifts. The difference would be that for flash lag, there's two separate stimuli. So there's a moving stimuli, and then there's a, moving stimulus, and there's a static stimulus, right? And the static stimulus is the one that's flashed very briefly as the moving stimulus moves across the screen. In the flash jump illusion, there's only one stimulus, which is the moving bar, and the color change occurs within that moving bar. So it's a problem of feature integration between the motion of the bar and then the color change that happens. So I guess that would be the difference, but they do fall under the same category of motion-induced position shifts.
Okay, do you have other questions? Harpreet, let me ask you the question that I asked the call before. What stage are you at with this work?
So we're still in the writing and drafting stage. I'm just finalizing how to present this with my supervisor and my co-authors, but we should be submitting this for publication soon.
That's great for a master's degree.
Yeah.
All right. Okay, I want to thank everyone again, and thank you for all the great questions. I think it was a nice and interesting discussion. Good luck, everyone, with publications and with the work, and see you later.
All right, thank you so much. Bye-bye.
Thank you. Bye-bye.
For joining in. We're going to give a little bit more time for people so that they can actually join in and tune into the talks here. You can see this beautiful splash screen that Saeed has created for us. He is joining me on the back end today. Thank you, Saeed, for being here with us. And we're going to be listening to talks from Bianca, Samantha, and Ella in that order. And I'm really excited about these talks. Some will be a little bit closer to what I actually do myself. Others will be a little bit further removed. But yeah, that's still really cool. I'm really looking forward to learning new things. So I think with that, we can start with Bianca's talk. Bianca Battaletto uses she, her pronouns, and she is currently a PhD in the Crawford Lab at York University in Toronto, which is in Canada. And she's going to be talking about how saccades influence object orientation versus identity. Feel free to take it away.
Thank you very much. Thank you for the introduction. And I would like to start off by saying thank you to the Neuromatch organizers. I'm really delighted to be giving this talk today. So without further ado, I would like to start off by introducing transsaccadic integration, which is known as the ability to perceive visual information, store that visual information, and then integrate the information across what are known as rapid eye movements, what we also call saccades. Now, the cortical mechanisms for transsaccadic integration are actually little known for object features. To date, object orientation has been investigated, and transacadic integration has been shown to be associated with activation in inferior as well as superior parietal cortex, in addition to extrastriate cortex within the occipital lobe. Now for spatial frequency, which is another object feature, transacadic processes have been shown to engage more posteromedial and superior parietal cortex, in addition to striate occipital cortex. So already we see that there are similarities, but some differences therein. And so this begs the question of how do cortical mechanisms for transacadic perception of different object features compare when they're tested directly? Now we boiled this down to a more specific question, which was, do the cortical correlates of transacadic perception differ for changes in object orientation versus shape? And our hypothesis was that changes in orientation versus shape occurring across saccades will engage differently various regions within occipital and parietal cortex. So how did we go about trying to answer this question? Well, we used a functional magnetic resonance imaging paradigm that looks somewhat like what I'm about to show you. In this example trial, participants started off by fixating on the left fixation cross for 7.5 seconds. Then an object was presented centrally for 3 seconds at one of 2 possible orientations. In this case, it was 45 degrees from horizontal. This was then followed by a 750 millisecond static noise mask. And after this, participants saw the fixation cross either continued fixating on the same initial fixation cross for our fixate condition, or they made a saccade to the other fixation cross position for 750 milliseconds. This was then followed by the presentation of an object. Now, it was either the same object presented at the orthogonal orientation for our same shape, different orientation condition, or it was a different object at the same initial orientation. So our different shape, same orientation condition. And these 2 main conditions were repeated for our fixate and for our saccade condition types. And the stimulus here was presented again for 3 seconds. This was finally followed by an instruction I or O, which was presented for 3 seconds. During this period, participants had the instruction to make a button press to indicate their judgment about whether the shape or the object had changed across these 2 stimulus presentations. So, and across the trials, we had our choice of these 3 stimuli. So a barrel-shaped object, a rectangle, and an hourglass-shaped object, to keep it more fun and exciting for our participants. So we had 17 participants, all of whom were right-handed. Their data were collected using the magnet that we have at York University, our 3 Tesla Siemens TIM Trio magnet. And once we collected their data, we analyzed it using a software program called Brain Voyager QX, version 20.6 for those interested. So what did we find? Well, first we looked at what changes in orientation do to the brain. So we looked at the subsample of trials during which orientation had changed across the stimulus presentations. And within those, we looked for an effect that was greater for saccades than for fixations. And that activation is shown here in this yellow-orange color. Now, this is an inflated brain rendering of the left hemisphere of an example participant. And it's the medial view because the medial view contains the most pertinent data for you. So the activation that we found for this condition was sequestered to this occipital lobe. So heavily centered on the cuneus as well as the lingual gyrus. What about the right hemisphere? Well, we have a similar story in that there's activation in occipital cortex again in this lingual gyrus. So if we take something away from these results, it's that when changes in orientation occur across saccades, they engage the occipital cortex. That was our first feature that we looked at. What about the other feature, which was change in shape? This time, we only focused on the subsample of trials during which shape had changed across saccades. And again, we're looking for an effect that's greater for when saccades occurred than when fixation occurred. Again, on this medial view of the left hemisphere, we see a lot of activation, again in occipital cortex and lingual gyrus. But interestingly, this activation penetrates into the parietal cortex, specifically in this precuneus area. And we have a very similar story in the right hemisphere in that this occipital activation is again centered on lingual gyrus, but again, it extends into parietal cortex and again, precuneus in this superior parietal lobule region. So if we have a summary for this particular feature, it's that when changes in shape occur across saccades, they not only engage the occipital cortex, but they also extend into parietal cortex. So now we come to our head-to-head comparison of these features. This time we looked within regions that show saccade-specific effects, so an effect greater for saccades than to fixations. And within those regions, we looked for an effect that was different or greater for orientation than for shape. So this orange activation shows actually a greater effect for when orientation changed across saccades than for shape. And what do we see? Well, somewhat unsurprisingly, I think, is that this activation is again sequestered to the occipital lobe, namely centered on this cuneus region. And we have very little effect in the right hemisphere. So when we compare our features directly, this head-to-head interaction, we see that these differences in the activation for changes in orientation versus shape that occur across saccades occur in occipital cortex. So it does seem to suggest that there are some differences here. So our conclusions are that we saw that when transsaccadic perception of orientation change occurred, this was activated or associated with occipital activation. When transsaccadic perception of shape change occurred, it was associated with activation in both occipital as well as parietal cortex. And when we looked at saccade-specific cortical regions, we saw that it was occipital cortex that was more strongly activated for changes in orientation than shape when we compared them directly. Now I have to point out that these are somewhat preliminary findings, but they're already pointing to similarities, but also a differential engagement of regions within occipital and parietal cortex for these transsaccadic mechanisms related, in this case, to object orientation versus shape. And so we need some further analysis to disentangle what is the underlying reason for these similarities, but also subtle differences for these object features. And with that, I'd like to thank the Crawford lab members, the MRI technologists who helped me collect these data, the funding agencies, my wonderful supervisor and co-authors, and thank you all very much for your time and attention.
Thank you so much, Bianca. That was awesome. So I got a question from Said, who was asking me to remind viewers that join us through YouTube that you can also use the chat there to ask any questions to Bianca and also to other speakers. So Bianca, could you pull up the paradigm for me again?
Of course.
I have a question about the saccade specifically.
Yes. Oh, geez. Okay.
Awesome. Yeah, thank you. So I was wondering if you have eye tracking data and whether it's the case that participants are actually moving their eyes from the point that they're fixating on the left hand side of the screen in this example and move it to the right, like in a particular way. Because I think what I'm getting at is that I can see how in the pursuits of the eye movement, it's of course much larger than a saccade. When I think of a saccade, I think more of well, maybe I'm just biased towards microsaccades here. But I was curious whether, yeah, that shift in like visual fields is going to be important for your research here, because yeah, I think I'm getting at, are there differences between visual cortex and parietal cortex in their receptive field sizes, and we know that's true. So how does that influence the findings here?
Well, that's a very good question, actually. So the change in visual field is something that we wanted to, what's the word I'm looking for? To really utilize. This was, by allowing us to do this, we're sort of getting at this was sort of based on like an adaptation effect. And so the adaptation type paradigms, we want to exploit something like a change in visual field. We're allowing us to see these changes occur across the hemispheres, across these visual fields, specifically for saccades. That's why, as you notice, this is a horizontal saccade, not a vertical. So that was actually that's a very good point, and it was important for us. Whether the size of the saccade and how that would be affecting the activation in the brain, I think that's also an important point. We wanted to, really, we were utilizing previous paradigms for this, and we saw that we didn't really want to allow very much vision of the object within the fovea. It's more of a peripheral design, as you can tell. And so I don't have a clear answer on that. Certainly there must be differences because of exactly what you said. There are receptive field size differences. However, I think that interestingly, we're seeing this activation in occipital cortex. And so what could be driving that? I mean, it is, early visual cortex, and those receptive field sizes aren't quite the size of the ones that you find in parietal cortex. So I don't have a very clever answer for you, but I do think that it is interesting, and we should definitely look at that a little bit further in terms of how it is that we're getting this occipital activation, given the fact that these, saccades are so large and the receptive field sizes are, much smaller in occipital cortex than in parietal cortex, for example. So I'll add that to my list of things to to think about.
Thank you so much, Priyanka. I got a question from Priyanka in the Q&A for you. She's asking, given that the subjects are getting 700 milliseconds to make the saccades, is this actually still transsaccadic, or is it more like post-saccadic?
That's another very good point, a valid point. I think individuals, my colleagues in the specifically transsaccadic integration aspect of the field would argue that it's not quite the integration. That's why I've called it more perception as opposed to integration, because it's not this, especially since we have a mask in there, right? It's not quite this instant integration of this information. I would still argue that it is transsaccadic perception because, that they are still making the saccade, and the bold signal that we're investigating does take all of this into account. It is a slow signal, and so that's why we wanted to make it as long and to allow people to make the saccade before they see the object. And we didn't want that to taint any of our eye movement data or traces or anything like that. So I would still argue that it is transsaccadic perception. I wouldn't argue that it is for sure transsaccadic integration.
Okay, thank you so much for that clarification, Bianca. Unfortunately, we have to move on, or actually, yay, we're moving on because Samantha is going to take it away for us. Thank you so much, Bianca. If you can answer questions that might pop up still in the chat, then please do so. I'm going to introduce Samantha. Samantha uses she, her pronouns, and she is a student in the Menard Lab at Queen's University, and she's going to be talking about the very important topic of the well-being of lab animals.
Thanks for the intro, Tara. My talk might be a bit of a change of pace with respect to applying more so to behavioral neuroscience employing animal models rather than pure cognition, but I'm excited to share these findings with you. So I'll just dive right in. So as many of you probably know, there is a long history of using rodents in neuroscience research. Now, a longstanding practice is to use basic standardized housing systems for laboratory animals. This has typically involved housing rodents either singly or in pairs in a cage that contains bedding and really nothing else. However, it's become more and more recognized that these barren housing conditions can have adverse effects on animals and their well-being. And because of this, it's become really common for many research facilities to supplement the animals' home cages with these things called environmental refinements, which frequently means providing species-appropriate items within the home cage, for example, nesting material, toys for chewing on or playing with, so the animals feel safe but engaged in that home cage environment. However, these refinements have been implemented without empirical validation of their effects on animals and their behavior. Now, without studies examining either of these factors, it's really unclear whether the refinement items actually do improve rodent welfare. Another critical issue concerns the reproducibility of research findings. It could be that these refinements might alter behavioral baselines, creating a divide between past and present research endeavors. So because of this, we designed a study to examine whether refinements in the home cages of laboratory rats actually do improve their well-being or influence behavioral baselines as indexed by well-validated animal models of anxiety and depression-related behavior. To this end, male rats were randomly assigned to either a refined condition or a standard housing condition. All rats were singly housed, but the cages of the rats in the refined condition contained nesting material, a hide tube, a wooden gnawing block, and a green nylon bone, which you can see on the bottom of my slide here. By contrast, however, rats in the standard housing condition were not exposed to any of these refinement items. Now, we predicted that rats in the refined condition would exhibit improved welfare as indexed by decreased anxiety-like and depression-like behavior, because that's why they've been so commonly implemented over the years. However, our results were really surprising in a lot of ways. The most startling findings we observed were in the novelty-induced suppression of feeding test, the NSFT, and the sucrose preference test. So I'm going to walk you through those ones today. Now, before I dive into the findings from the NSFT, because I don't know how familiar you are with animal research, I will give you a quick run through of the test and the results we typically see. In this test, the experimenter measures rats' latencies to consume a novel treat, for example, a graham cracker, in both a familiar home cage and a novel environment. And in this test, longer latencies to begin eating the novel treat or graham cracker due to the novelty component are interpreted as heightened anxiety-like behavior. So on habituation day 1, which you can see on the left of my slide here, rats are first presented with that novel treat in their familiar home cage. Here, it's typical for rats to display latencies of around 160 seconds. However, with repeated trials in the familiar home cages on habituation days 2 through 4, and then again on that home cage test, we see rats gradually decrease their latencies to as little as 50 seconds. On the novel test day, however, rats are tested in a novel cage in a novel room, and again presented with that now familiar graham cracker. Here it's typical for rats to again display prolonged latencies of around that 160-second mark, because again, we have that added novelty component just based on that unfamiliar testing environment. Now, these behavioral predictions are in line with other literature in the field, as well as work conducted previously by other students in our lab. However, this is the behavior that we actually saw when we collapsed latencies across both housing conditions. So across all testing days, we can see that rats displayed significantly higher latencies to initiate that snack consumption than we predicted. For instance, we can see that on habituation day 1, all of the rats took approximately 310 seconds, and these latencies increased really intensely to 470 seconds during that novel cage test. This is really unusual and suggests that the rats were showing very high levels of anxiety-like behavior compared to what we would normally predict. Now, another unusual finding that you can see now that we're comparing rats between both the standard and the refined condition concerns the rats' habituation profiles. Here, the standard housed rats actually displayed the expected decreases in latencies across those trials that I mentioned previously. However, this typical habituation curve was not shown by the refined rats, and this actually led to significant between group differences on habituations day 3 and 4. So if you're, oh, sorry, I think I'm actually showing you the wrong screen still. There you go. Can you see that now? Sorry, I got ahead of myself. So if you're following me now, you can see that these were actually the exact opposite of the results that we were expecting to see, with those refined housed rats faring worse in terms of their heightened anxiety-like behavior. This unexpected pattern of behavior was actually observed again in the next test that I'll be discussing, the sucrose preference test. Okay, make sure I have the right slide this time. So the sucrose preference test is a commonly used animal model of actually depression-like behavior. And this test measures anhedonic-like deficits in reward processing, a core symptom of depression in humans. And in the SPT or sucrose preference test, rats are presented with 2 bottles, one containing plain drinking water, the other rewarding sucrose solution. Healthy rodents do possess an innate interest in sweet solutions, hence they typically demonstrate a bias towards the sucrose. However, lack of preference for the sucrose is interpreted as a display of anhedonic or depression-like behavior. And the way we assess sucrose preference is by calculating the ratio of sucrose consumption relative to the plain drinking water, which is their sucrose preference score. So to establish whether our animals were displaying anhedonic-like behavior, we evaluated each group's mean sucrose preference score against the conventional norms in the literature. For instance, sucrose preference scores below 65% are typically interpreted as anhedonic-like, whereas being above 80% is typically, it's conventional for healthier non-stressed rodents. So the dotted lines that I have cutting across the graph here are indicating the 65% and the 80% cutoff values. Now, again, interestingly, and in contrast to our original hypothesis, the refined housed rats again fared worse in terms of their well-being and worsened depression-related behavior relative to those standard housed rats. Refinement rats displayed sucrose preference scores consistent with those that are designated as being anhedonic-like, whereas the rats in the standard housing condition displayed preference scores that were significantly above that 65% cutoff. When we repeated our analysis using the 80% threshold value, or what is considered healthy, we found that the standard housed rats did not differ significantly from this value, whereas the refined housed rats fell significantly below the 80% cutoff. So this led us to conclude that the refined housed rats might be more susceptible to depression or anhedonic-like behavior. Okay, so I was going to quickly rehash the results, but I'll just jump ahead. Basically what we think, because the rats were showing heightened anhedonic-like behavior in both tests, we think that the refinement items in and of themselves may have contributed to an anhedonic-like state within the refined housed rats. This was a bit of a head scratcher for us because they've been implemented so widely in the interest of animal well-being, but we think that they might have contributed to overcrowding within the home cage, which could have worsen their depression-like phenotype. There is evidence within the field to support this hypothesis, as overcrowding can lead to heightened depression-like behavior. However, it doesn't really account for the heightened anxiety-like behavior that we saw by all rats in the novelty-induced suppression of feeding. Upon much reflection, we think that this might be caused by the novel use of independently ventilated cages. Traditionally, we've used ambient ventilation in our laboratory colonies, which you can see on the left side of the screen here, through which the air can flow freely in and out of the cage. By contrast, in this new housing system, the independently ventilated cages are hermetically sealed. And this is really good for transgenic animals or immune-compromised animals, but it basically, in singly housed animals, it contributes to complete social isolation. Now we know by excess literature within the field of neuroscience that social isolation in social animals can contribute to chronic stress and really exacerbate any anxiety or depression-like phenotype. So we believe that within the current study, the rats in both housing conditions were under chronic stress due to complete social isolation caused by the IVCs. And we think that this was then compounded further within the refined housing group due to overcrowding by way of refinement items within the home cage. So in summary, it's really unclear whether these refinements actually do improve the well-being of animals used in research, although the current results do suggest that the crowding by way of refinement items might increase a depression-like phenotype. This finding was potentially confounded by other inadvertent stressors within the home colony, such as these IVCs. It's really crucial that we further understand how these minor changes to the home cage environment can impact laboratory animals' behavior, both to advance the welfare of animals used in neuroscience research and also to ensure that any research efforts within our field aren't being wasted. So that's all that I have. Thanks for listening. I'll be happy to answer any of your questions.
Thank you so much, Samantha. That was really interesting. I'm very happy that you explained a little bit about these IVCs at the end of your talk, because I was getting pretty worried about how we keep rats in the lab. So one thing that I was wondering is, as these IVCs were probably applied to all the rats that participated as subjects in this study, that can't really explain why you see these heightened anxiety behaviors in those rats that had actually these enriched cages.
Yeah, so that it's actually a really interesting phenomenon, and it's opened up a new line of research for our lab. Actually, a lot of the undergraduate students that are coming in now are directly comparing the use of the open top cages compared to the IVCs, because within the scope of our study, we really couldn't definitively say one way or the other, this is the cause or not, because we weren't comparing those variables. But our lab now is actually, we're traditionally an anxiety lab, and we look more so at neurodevelopment actually, but it's opened up this new line of inquiry where we're trying to parse out how these different variables, whether it be the caging, the bedding, even the lighting within the home cage system, can influence different behavioral baselines. So I don't know if, does that kind of answer your question?
Yeah, no, it does. Thank you so much. So relatedly, is that then where this research is going to go? Or is it something that you might say, well, let's compare other rats? Because I'm guessing that these rats that you tested now in these different, in the enriched and in the non-enriched cages are coming from the same type of animal, let's say. Is that what you're going to change next, or is it really sticking with the focus on these different cages?
So I believe that the scope of so I'm no longer involved in this project. This was my honors thesis from 2 years ago. But right now what's happening is they are looking more so just at those caging systems. So whether it has to do with the social isolation component of being in a cage that's hermetically sealed, like those independently ventilated cages, versus the ones that do have the open wire top. I kind of had to hustle a little bit at the end there, so I maybe couldn't really describe so well the differences between the two caging systems. But we really believe that what's contributing to it is just that inability to hear or really smell any of the other rats nearby in the colony room. So that is the variable that we're looking at most intensely for sure.
Okay, thank you so much for clarifying that. I appreciate it. I think in the interest of time, we're going to move on to our next speaker. Thanks again, Samantha.
Thank you.
You're up next. So our next speaker is Ella Potwalny, and she uses she, her pronouns. She's currently a postdoc at NYU in the lab of Bayu He, and she's going to talk about her recently published MEG study where she's linking pupil size to arousal. Bring it on. Samantha, you have to stop sharing your screen for...
Oh, sorry.
No worries.
There.
All right.
Perfect, thank you.
Now I should be able to. All right, should be on. Hi everyone, thank you very much for joining us today, and I'm very happy to talk about this work about arousal states and how they shape perceptual decisions. Actually, this everything, all the results here that I'm going to present are new. They were not included in the paper that was published last year. So, yeah, this manuscript will be out soon. But if you are interested, please email me and I will send you the manuscript. Yeah, and this work was done with Lina King and Bu He. So arousal state is a very powerful predictor of human and animal behavior.
And this is especially evident, of course, when we look at the extreme arousal states, such as a deep sleep, or extremely active behavior, such as fight or flight response. And this type of behaviors modulated by brainstem neuromodulatory systems, which also affect the pupil size. So the pupil size gets larger with heightened arousal state. And also there are many projections from brainstem to cortex. So neuromodulation also affects the frequency content of cortical population activity. And with higher arousal, we have more higher frequency fluctuations. And of course, there is a lot going on in between. In intermediate arousal states, our pupils still fluctuate all the time, and there are changes in frequency content of cortical population activity. And interestingly, all this happens even when there is no stimulus present at all and when we don't move at all. And this kind of states we usually refer to as tonic arousal. And this is what I'm going to focus on today. So more specifically, I'm going to show you some results of our recent investigation into the question of how spontaneous variation in tonic arousal influence future decisions and how arousal-linked cortical spectral power is involved in this process. So to address this question, I recorded magnetoencephalography, MEG activity in human participants, and I use eye tracker to record the pupil size. And this was done in both resting state and in perceptual decision-making task. So the first thing that I could see that using resting state and also pre-stimulus baseline, where only the participants needed only to fixate on a cross, I was able to calculate the spectral power in all frequency bands across the whole brain and to see how it changes together with pupil size. So here we already see this gradient that we expected in delta band, where we have decrease of this low frequency activity together with arousal and increase in gamma activity with arousal. And there is something more interesting going on here in between. And to quantify this better, I use an atlas of resting state networks. And for each network, I calculated the mapping between the power and the pupil size. So, and here we can see, looking at it more closely, we can see that there is this interesting inverted U-shaped relationship with pupil size and alpha power. And I'm going to use now these mappings between pupil-linked arousal and cortical spectral power to understand how they affect behavior. Now, my very simple assumption of what is state-dependent behavior is to say that behavior is a function of a stimulus that is presented and a state before stimulus onset, the state of the brain. Now, to make this very simple, the state fluctuates, as we said, this tonic arousal state fluctuates spontaneously, even without any stimulus present. So this I cannot control, but I can keep the stimulus constant. So when I keep the stimulus constant, making the link between the state to behavior would be easier. To understand how the state influences behavior, I will also look into the shared contribution of pupil linked arousal, of the correlation between pupil size and the spectral power, but also I can think about additional neural mechanisms that regulate the pupil size, and also there are additional neural mechanisms unrelated to arousal that regulate the spectral power in cortical population activity. So I'm going to dissociate between all this. Right, so here is my task. So I use object detection task where the subjects need to report whether an object is present in the image and what is the object category. And before this main experiments begins, I want to identify the image contrast such that sometimes the subjects detect an object and sometimes they don't. So in the main experiment, in the end, there is on every trial I have exactly the same contrast, and it looks like some kind of foggy object. And sometimes the subjects say, "Oh yeah, I see it. I know what that is," and sometimes they don't, even though the stimulus is the same. And in addition to real objects, I also use scrambled images that preserve the low-level details, but they destroy the object itself. So I use this to quantify the false alarms. And yeah, to analyze behavior in this task, I quantified the reaction times and the accuracy of the categorization task, but also I calculate the decision detection criterion and sensitivity. So the more positive criterion is, the more conservative is the detection. Is the decision about whether there was an object or not. And the higher is with higher sensitivity, the subjects are just more readily can distinguish between signals and noise. So yeah, now I'm showing you the how these 4 behavioral metrics change together with pre-stimulus state. So what we see here is that with larger pupil size before stimulus onset, we have more liberal criterion, we have faster reaction times, we have increased sensitivity and increased categorization accuracy. Now, this is really interesting because even before the stimulus is presented, we can already say a lot about future behavior. I was interested to see if this pupil-linked effect can be explained by these changes in cortical power, also in the spectral power. So to do so, I basically regress out all the pupil size that I could predict from changes in spectral power across the whole cortex. And I see that the effect of pupil size on criterion and sensitivity disappears. So what this means is that these effects are very likely to be mediated, in fact, by cortical spectral power, and the effects on response time and category accuracy are attenuated. So what I was interested in next is to try to understand how residual spectral power, which is unrelated to arousal in different brain regions, and how it also, if it has additional effects on my behavioral metrics. So to do so, I again regressed out everything that is related to pupil. And what I saw is that pre-stimulus delta power in dorsal attention network that includes frontal eye field and IPS and intraparietal sulcus and also default mode networks, these regions have additional effect on decision criterion. And this is actually mirrors results in monkeys. And also I found that pre-stimulus alpha power in visual areas negatively correlates with sensitivity. And I'll skip that. And finally, I just wanted to briefly mention that we also looked at spontaneous events of dilation and constriction. And we found this beautiful event-related activity and MEG signal, which was very similar to what was found in monkey LC, but we couldn't find any behavioral effects of this dilation and constriction events. So it doesn't matter if you present a stimulus during a dilation or a constriction. And yeah, this is the summary of my finding. It's really, really interesting to see how changes of arousal when nothing happens before stimulus onset, how powerful they can be to predict future behavior.
Great, Ella, that was really interesting. Thank you so much for your talk. So I have a question about how long the time period is in which you actually estimate the pupil size and also, of course, the spectral power.
2 seconds before stimulus onset. And actually we had, this task was designed specifically with understanding spontaneous activity in mind. And we made really long pre-stimulus intervals. So the pre-stimulus intervals were between 3 to 6 seconds. And this is very long for this kind of tasks. So we had wanted to have lots of time to make sure that, yeah.
No, that makes a lot of sense, because of course these changes are much slower than what we typically test in the lab, which actually kind of leads into my next question. So in outside the lab, how relevant do you think these findings are? Like, do you think this is something that's also influences our behavior in our everyday life, let's say?
Definitely. I think, brain is this large scale system where and arousal state is never stable. So there are things that going on all the time. And whether you have a stimulus that comes to you in one moment, whether it comes in another moment, your decision might be different just because the state of the brain is different. And it's not only about the decisions, it's about also perceptual experience. And we even think that false alarms in our task, they're not necessarily just I mistakenly press the wrong button, but it could be also false perception. Like if you walk around in the fog and visibility is very low, sometimes you can see something that is not there. So I think this future understanding of how the state of the brain shapes decisions and perception is very relevant for real life. Yeah.
Cool. Yeah, it was a really comprehensive talk. I really enjoyed that. Thank you so much. I think with that, we're getting to the end of this session. Thank you so much to all the speakers, and thank you for joining us. This was really a lot of fun. I learned a lot.
Thank you.
Thank you.
All. Take good care.
Thank you, everyone. Thank you. And goodbye. Bye-bye.